ENSA and Semiorto as the companies, Cajamar, TETAEX, Unitus, and University of Taiwan. So the idea of the objective of this whole package is to introduce stress tolerance that the public partners have been identified in the previous research, and introduce that tolerance into elite materials that are provided by ENSA and Semiorto. There are three major objectives. First, to define the strategy to combine tolerant non-elite variability to elite. Then the introduction of this non-elite variability into elite lines to develop lines, and also we're working with root stocks. And finally, assess the material that we will develop in the project in different working conditions and pedoclimatic areas. We have 7 tasks in this work package, and I'm going to summarize all of them one by one in the next slides. The first is catalog unification of resources and information available for improving tomato resilience to climate change. So this task is to gather all the information that we have for previous QTL mapping done for the groups and also in the literature about QTLs involving drought, temperature, and salinity stress, and also stigma position, I forgot to mention here, and to be part of the OneStore repository that is being developed in the work package 3. For doing this, during the first year, we established a criteria to choose the works that will be included in this first catalog. So we use only QTLs from mapping populations where the QTLs had a physical position. So we are not working with QTLs that only refers to genetic position, because we cannot compare the position of the QTLs between works, or it's difficult to compare. It's not straightforward. And initially, we focus in traits related directly with the stress response, so physiological traits or reproductive traits. And with this criteria, we selected 10 mapping populations. And from these 10 mapping populations, we detected 104 QTLs for heat tolerance, 19 for salt tolerance, and 39 for drought tolerance. Here you have the map or the position of these QTLs, see, with a color code, red for heat tolerance, blue for salt tolerance, green for drought tolerance. And here you see the position of the QTL that we have integrated in this catalog in the tomato genome. And clearly there is not a random distribution of the QTLs in the genome. We have 1, 2, 3, 4, 5 hotspots of QTLs, and in some of the QTLs of these hotspots, there are QTLs for different stresses. So I think this is a nice results to dig in and to see maybe same QTL is responsible to respond to more than one stress. We had a deliverable for this task on month 12, and this deliverable was submitted on time. And let's see what the reviewers say. And for the next steps for this catalog, we have to include other works that they did GWAS analysis. This work were not included in this catalog, but we included in the catalog for the database. We have to add new experiments. There, our group and other groups from the consortium, they are mapping new QTLs with different populations. So we will include all of these new experiments and any other information that will appear in the bibliography, and we'll integrate all this catalog in the database in collaboration with Work Package 3. The second task is the assessment of response to different stresses of not-elite tolerant breeding lines. So the different partners are providing different lines with response to different stresses. So the initial idea is to share all these lines between the partners, and all these lines will be evaluated for draw, heat, and salt to see if they have different response to the different stresses. So this is the summary of the lines that we want to share. So in PESIC, we have 2 heat tolerance lines. This is the money-maker control, and we have these are 2 international lines from Pimpinelli folium, with one in chromosome 1, another chromosome 12. That in previous project we saw that they have a high heat tolerance. This one with introduction chromosome 11 has a high tolerance to Ploss and Enroth. ENSA had a number of hybrids that we saw in previous work also that they had a high heat tolerance. IVIA develops population of reels also in across with Pimpinelli folium, and they were working with this population for a lot of years, and they found several of the reels that they were very good rootstocks for salt tolerance. So these reels will be used as rootstock source. There are some lines also that Nina found with a high heat tolerance, and Unitus is working in extreme position. I found a number of these are traditional European or Mediterranean varieties from the Triton project with different phenotype for the extreme position, and also these 3 are 3 QTLs that, as I will show you later, they map for extreme position. So they are developing near lines for these 3 QTLs. From Taiwan, from Taiwan, CAI analyze a reel population, is an interspecific population from one sensitive variety and one tolerant variety. And in this population, we map a QTL in chromosome 3 for pollen viability at high temperatures. We'll see later also more details on this QTL. And one of the reels that contains this QTL is the reel 41. So this reel 41 also will be included in the basic germplasm for this for package. And Uji Dani provide 3 builds and the hybrids that they have a high drought tolerance. So this, so we also include this. So the first idea was to share all the seeds, and we thought that the best way to do it is to centralize the seed distribution by PESIC. So it will be, in principle, the easiest way to do it, and it turned out to be the most difficult thing to do it because we had a phytosanitary problem. We found that some of our seed stocks are infected by a virus, the tomato mottle mosaic virus, that is not in the list of the virus that you have to test to share seeds, but obviously we cannot distribute seeds we know that are already infected with this virus. The local authorities are aware of that, so at this point we are banned to share any seed. We cannot distribute any seed in this moment, so we couldn't distribute the seed with the partners. Now we're working on that. We are revising our protocol for seed disinfection, and now we are getting clean seed, but we have to develop new seed from clean plants to distribute. So this first objective that we have for this year, we couldn't achieve it because this problem with phytosanitary problem, and also the other party, they have a lot of problem with the bureaucracy because this bureaucracy has changed, and for is sometimes the authorities don't help to speed up the bureaucracy, and there were some problems. Danny managed to send the seeds, his seeds. So we received the seeds from Israel, and these lines are evaluated by different parents, as you will see later. So the lines that we could evaluate here in CSIC include traditional lines from Unitus that we had already in our premise, and the ones from Israel, some from ENSA, and some that we had already here. So we could reevaluate those seeds. And what we do, our experimental setup is we grow the plants in normal conditions. Normal condition is in a greenhouse with temperature control. So the plants are grown with 25 Celsius during the day and 20 degrees at night. So we score the number of flowers, the number of fruits, and the percentage of fruit set in the second and the third thrust. And then when we score this, we clean all the plants, we change the temperature, and we increase to 30 during the day and 25 during the night. These are minimum temperatures. During the day, sometimes we have higher, of course. And after 4 weeks, we measure number of flowers per truss, number of fruits that are set, and the percentage of fruit set. We remove all the branches, clean all the plants again. We increase the temperature to 35 to 30 during the day, to 30 during the night. And after 4 weeks, we measure the flower and the fruit set. The results, the molecular results are shown at the third temperature, and this is the results that you can see here. We separate the intermediate with determinate varieties because the behavior is different. And this is the fruit set of Moneymaker at the third temperature, and you see it's very low. Usually it's around between, I mean, the mean is around 5%, 10%, not more. And we found the two varieties from ENSA. Actually, they were very high tolerant. The reel from Taiwan is also is quite tolerant. This is one of our reels that had some tolerance in the past. In this experiment, didn't show a good tolerance, even though there are some plants that were very tolerant, but it was more variability this year than we had the year before. So maybe that's why we couldn't see the tolerance in this integration line. And for the traditional tomatoes, only one, but it's interesting. We didn't expect that. So a good heat tolerance. Regarding the determinates are the ones that Danny Simon sent us. So the control is M82. In general, the determinates they have better tolerance than the indeterminates. Anyway, one of the builds show a significant higher heat tolerance. So this build apparently is tolerant for draw, but also for heat. These builds come from this population that Daniel already introduced you yesterday, this build population. And from this build population is very good, very nice to detect QTLs, but also to detect interaction between QTLs, because as you have a lot of lines, a lot of variability, it has a lot of power to study interaction between QTLs. That is one of the things that usually we cannot handle in the regular QTL mapping experiments. So for all the in this lower part, here in the upper part will be the QTS detected by chromosomes. So the the yellow regions are regions where are QTLs. We will say minor effect QTLs, and here you see some yellow pale blue lines are the interactions between two regions. Okay. So from all this plot of interaction, he shows one that interaction between chromosome 1 and chromosome 7. This is the control iteration line. This is the control. This is the lines that have only one of the QTLs. This is the line that have the other QTL. So you see that there is no effect on total yield, but the lines that have both QTLs, they have a significant higher yield. This is for total yield, and this is for Briggs yield combination. And so these are the lines that we are using in Harvester. Andrea for Unitus, he selected a number of traditional tomatoes with different phenotype for stigma position. So there are 2 inserted, 2 exerted, and some that, according to his data, is heat sensitive. But we found that this one is heat tolerant. So it's something that we can work on it because I think it's interesting if we have a new heat tolerant source. So we, as I told you, Andrea wanted to share all these plans with all this material with us, but not shipping permits didn't arrive on time. So for last year was not possible. Fortunately, some of us will have already this live, so we could test them. And also the problem that there's material that is still stuck in CSIC, so he couldn't work with this material. Unina, they are working with Semi-Orto, and they had a number of lines that selected previously for good performance under stress, and also they got the bill from UTI. The plants were transplanted in June to ensure high tolerance. They do the experiment in fields and the plastic tunnels. So now they don't have the whole data, but when they send this slide, they are still harvesting and analyzing the data. But they found nice variability for total yield among these lines, and also the preliminary data shows that the bills are also heat tolerant. So we have to evidence that these bills also they have a good heat tolerance. Semi-Orto also could work with 4 of the traditional varieties from Traditom that were selected by Andrea, and they could verify the phenotype for the eczema position. So what we have to do for next month, we need to solve this the phyto problems that we have. We already have the clean plants in the greenhouse. We check with RT-PCR the virus several times. We have a master on virus detection now, and in seed disinfection. So I will, if you are interested, I can share with you our protocol for seed disinfection. And but the plants are now in the greenhouse, and we need to collect the seeds. and then do all the bureaucracy and to convince our authority that we are virus free and we can share seeds. So when we solve this problem, we hope we will do it on time. We'll send the seeds to the different partners and do what we couldn't do the last year. So let's check the salt tolerance, the drought tolerance, the heat stress, and the final position. UNIT and Semi-Orto, they are working together, so they will keep evaluating the materials and the different materials are developing in the project. The tax 3, 6.3 is the validation of strategies to increase the stress tolerance of the material. So the idea of this task is we have the QTLs that have effect in the background that we use, we studied before, but we don't know if they will have an effect in the elite background. So we need to verify the effect of the QTLs in the elite background. Also, the QTLs usually are mapped in a very long region, so this is not useful or it's not easy to work with that for fast molecular acid selection. So we are going to find up some of the QTLs to get markers closer to the QTLs and facilitate the marker acid selection. And also, when we want to use some root stocks and test some of the materials as root stock for draw salt tolerance, salt and bluesol tolerance. Here in PESIC and also with Cajamar, every time I say PESIC, I have to tell you, every time PESIC, it means PESIC plus Cajamar because we do all the experiments in the facilities. Okay. So one of our objectives last year was to find up the heat tolerance QTL that is in international line SP14, but because of this problem with the virus, we couldn't transfer the seedlings to the greenhouse, so we have to postpone to next year. So we will repeat the experiment. And for root stock, we had 3 different genotypes with different behavior for blossom error incidence. Moneymaker, you know Moneymaker, so Moneymaker you have usually incidence of blossom enrod. This interaction line, SP11.4, is highly tolerant for blossom enrod. We detected this interaction line in the Tongen project, and when Moneymaker had heavy blossom enrod, this line was clean. The fruits were very, very clean. And this one is the opposite. This was shared with us by Esther Vandernap, and it's the opposite. So it's highly sensitive to blossom enrod. The fruits are, this is a kind of Theraciforme very from America, so small fruits, but the fruits are absolutely black when they have incidence. It's not big. It's the whole fruit is blossoming. It's terrible. And with this, so these are the so we use 2 rootstock, one rootstock control, Moneymaker, and the other rootstock, the resistant interaction line. This year we didn't have a high blossom enrod incidence. Here you see the different. This is the interaction line. You can see the mean that it has no blossom enrod, nearly no blossom enrod at all. And Moneymaker has is around 20% of the fruits. That is low compared with other years. Still, we see the differences, but they are not as high as we expected. So it's difficult to make conclusions with this low incidence. This is the incidence of the line from Esther, and you see that the incidence of enrod is very, 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 very high, nearly 80%. And here you can see also the effect of the rootstock. When rootstock is Moneymaker, the incidence is 100%. This is the incidence in 2 different temperatures, control and heat stress. And when we use the resistance line a rootstock, the integer is lower. So there is clearly an effect of the rootstock in the error incidence. We try to find the QTL or the gene for blossom error tolerance. In our previous project, the gene was mapped at the end of 2011 in interval that is about 1 megabase. And we developed a number of recombinants in this region. We evaluate these recombinants in this year, but as I told you, the incidence was not high. The results are not as clear as we expected. And we found that this region here, in this part of the chromosome of the interaction, is the region with higher significance to blossom error tolerance. In our previous experiments, the QTL was in this part of the interaction, so we overlap in our new experiment of this year, we overlap the position, so it's not 100% clear, but well, there is evidence that the position of the gene is in this part of the integration. For validation, ENSA is doing crosses between eight different elite lines for determinate lines. They already crossed some of the lines with some of these lines with some of the interaction lines that they had already because of the Tongen project. So we have already processed with these lines and these with the ones from Israel, and they are waiting for us to send the line for Rosenrod and the new lines that Andrea is developing for stigma position. But some is going on. Some of the processes are going on. UNITO that is working with stigma position QTLs. They have 3 different populations. One interspecific population with pimpinellifolium as one of the parents, interspecific population, and a number of land races with contrasting phenotype to try to find markers by GWAS analysis. For the interspecific population, they already map 3 QTLs, and these QTLs are verified, and they are developing near lines for these 3 QTLs, and they started work. Last year they were phenotyping the land races for extreme position. The data that we have now are preliminary data, so the first broad phenotype of the extreme position. So, what is the next task for this task 6.3? We continue with the fine mapping of the heat tolerant PTL in SP14, and we keep working on the blossom error of 11.4. We're going to evaluate the hybrids that ENSA is doing, crossing the heat tolerant donors and the elite germplants. IVIA is going to check the solder of several rootstock, including the rootstock that they have developed in a common excision. ENSA will work to do needs to do the hybrids with Bertoller as soon as we send the sys to them. And they also generate F2 population from this process for QTL verification. And they are also interested in the next months to use the builds as root stock for draw tolerance. Unitus will do a fine phenotype for estimate of the land races, and after genotyping, they will look for association, and start the study of the other interspecies population between Catalone and Marmande. And Danny Produki will do the fine mapping of the QTLs that are in the builds in chromosome 7 and chromosome 1. So we will be able to manage better these QTLs. Tax 6.4, efficient management of previous defined QTLs and mapping new QTLs from new genotypes. This tax is performed by Kai from Taiwan University because as partner, he needs to have a specific task for him to get the funding from his government. So he has two different objectives. One is QTL mapping for heat tolerance in a new real population, and the fine mapping and development of version line for heat tolerance of pollen viability on chromosome 3. This is the real 41 that we use in the to introgress into the young plants. For the new reels, they have a repopulation by crossing a heat tolerance inbreeding lines, T star, and a heat tolerant inbreeding line that is Siberia. The magnet population consists of 176 reels, and these reels have been genotyped by rat capture. And the phenotyping is similar what we do regarding flower, fruit number, fruit set, and also pollen viability. So in spring, they evaluate these reels, and this summer they started developing this again the reels, and they are still analyzing this data. We don't have the data, but we have some data from the spring experiment, and next year they will evaluate again the reel during the summer. This is the temperatures that they have in Taiwan at soil and air. You see that the maximum temperatures are really high, and in this part of the summer it's impossible to grow any tomatoes because the soil temperatures are extremely high. So they have some moment of heavy rain that especially in the spring, it poses a problem to evaluate the plants. This is the segregation of the reels for fruit set, fruit numbers, in different traits related to reproductive traits. And the second objective is to fine map the QTL for pollen viability in chromosome 3. So they were setting up the molecular resistance selection system. So they set up the markers that flank the QTL, and now they have two reels, the reel 41, as famous reel now, and this one that is that is I didn't tell you, actually they found two QTLs, chromosome 10 and chromosome 3, so they interact, 3 and 10, and in this combination, homozygous CA and heterozygous, you get better heat tolerance. So that's why we are using this 41 to get to find out the QTL. And then they are using the other combination for the leading chromosome 11. Sorry, 10. But well, they are working on now on the fine mapping of this QTL. So for next month in summer they will evaluate the real population again, and when we finish the population, we have 3 years of 3 experiments of evaluation. So they will perform the QTL analysis, and they will screen recombinant plants to pursue the possible cloning of the QTL for pollen viability in chromosome 3. The task 6.5 is pyrimidine multiple stress tolerance, so combine different QTLs for different stresses, and also to make crosses between different rootstocks. Yeah, and crosses between different reels that they have salt tolerance to see if we have some heterosis. So in ENSA here in CSIC, we are crossing the heat tolerance sources with bare Brosen-Enroth and draw tolerant sources. Here they are crossing the salt tolerance and draw tolerant reels to combine both also combine both tolerance in one rootstock. So the next task 6.6, introduction of QTLs into elite germ plants. We decide to split the QTLs with the two companies. So ENSA will work with QTL that we detected the previous project Tom from CSIC and Taiwan, and also the QTLs from UJI, and Semiorto will work with the QTLs, the heat tolerance of QTLs from Nina and Unitos, and they may also be interested in the QTL from the draw tolerance. And they will perform several rounds of market selection to introduce these QTLs. So this already, this as I showed you already before, so the process that ENSA did to introduce the QTLs into the elite material. So the next task is we need to select the SNP markers for market selection. So we need to have the genotyping data for the elite material and the donors to make the market selection panel and the marker whether to use. And Semiorto with Nina also, they are doing something similar, and they will prepare the protocol to introduce the QTLs by market assisted selection. And the task 7.6.7, evaluation of advanced materials, different areas. Obviously, this is the task for the last year, so we didn't do anything on that. As soon as we have material that could be evaluated, so we will start evaluating this material. This is for the last year. Deviations, well, the major deviation is the limitation of system exchange. So because this phytosanitary issues that I told you before, and also some fine mapping that we couldn't do it because of this problem. So this problem has delay these activities at the breeding program, so we are going to have a delay, obviously, in some of the objectives that we have in this work package. Deliverables, the deliverables I told you before for the survey of QTLs is already submitted on time, and for the strategy, as we have these delays, we propose to delay this deliverable 2 months 30. Hope by then everything will resolve and we'll have a nice strategy to increase access tolerance into elite material. I think I'm too fast. And regarding dissemination and training activity, and dissemination, we have already two papers published, and I know that there are a few more that are coming. And I think this is all. Side, any question, comment? Let's discuss.
I may have a question is in relation with the QTL mapping that shows co-localization or tolerance to several stresses, and also shocks me that variety that is called Siberia is heat tolerant. So it's counterintuitive. So maybe this, you know, if this Siberia is also tolerant to cold.
I don't know. Kai, you know anything about the Siberia, or Peter? You can.
Oh yes, Siberian is also tolerance to cold. Yeah, yes. I think Peter can explain more. Yes, please.
Maybe one question, Tony, from Mathilde. Hello. Yes, I saw that you are assessing blossom and drought in heat stress, not in drought stress.
Exactly.
Which is surprising because usually we see a lot of blossom and drought in drought stress more than in heat stress.
Yeah, we do in heat stress because we do experiments in heat stress.
And you see some blossom in heat stress.
We see a lot of blossom this year, not as much, but in the last year we saw a lot of. We did also a small experiment in trying to induce blossom applying some drought stress also this year, but we couldn't get any blossom incident. So we try to induce in a regular greenhouse.
And just a remark also, we have published in a previous project several GWAS work and work on the magic population for several stresses, water stress, heat stress, salt stress. So if you need those data for the task 1, they are all available. And if you want some materials, we identified some lines which were tolerant to heat stress and water stress. And we have the phytosanitary passport now for all these lines. So if you are willing, some of them are available.
We can test some of them, yeah. Let's see how they behave in our experimental setup. Yeah. It's very interesting. Thank you, Mathilde. David?
Yeah, good morning. So I have a question about the blossom end rot. And it was at the beginning. I'm pretty sure that you know that beginning of this year, it was published a paper about the localization of blossom end rot. So they also established a QTL on chromosome 11. So I'm curious about if you did check if the population that or the localization that they found is correlated, have some correlation with the region that you found or not? And if yes, they also found 2 more QTLs that are related with the blossom end rot. So if are you thinking into introduce those QTLs in your study or not?
Yeah, we are aware of that because this work is done by Esther Vandernap. I have a very fluent relationship with Esther, and we had a conversation 1 year ago when we found out that they were working also with blossom end rot. Actually, the sensitive accession was sent by her. So this sensitive accession that I showed you is the one that she used for mapping. They didn't map the tolerance, they mapped the sensitivity, because they mapped this accession that was very sensitive. So it's a complementary, but we are now checking his mapping results, our mapping results, and it's possible that we are working with the same people. And we are talking with her, and we are going to with her to see, try to collaborate with them. And I think we will be able to use it because I've been collaborating with Esther for the last 20 years, so it shouldn't be a problem. And I see, I know that there are another QTLs, and but in our case, what we know is only with the chromosome 11 we have tolerance. We don't need other QTL. Yeah, the one that we have by itself alone is enough to have tolerance to blossom end rot. It doesn't mean that there are other, of course, there may be other QTLs, but this one by itself is sufficient to get tolerance. So yes, the next weeks we will arrange a meeting with Esther and share the results and see how we can collaborate to increase our knowledge. Yeah.
Okay, thank you.
Thank you.
Any other question in relation to the work package 6 on climate change for Tony or for any other of the participants on this work package?
Morning, I'm Rafa from Malaga.
Hola.
I don't know, my camera is not working well, but anyway, this is an answer for Mathilde. Mathilde, this was done in greenhouse, and in greenhouse, heat and drought is almost the same, because at the midday, you know, plants are submitted to a very demand of transpiration. So plants cannot cope with this transpiration, and then it's just like a drought stress.
Yes, but you can irrigate them.
Yeah, but it's regardless the
Irrigation.
Yeah, it's regardless. Even with, you know, plenty of water in the soil, plants cannot cope with the transpiration rate.
Yeah.
So fruits at the end are competing for water with the leaves in transpiration. So fruits are submitted to drought. So that's why it's very common to have bare blossom end rot, even if the conditions in the soils are okay.
I think it's also intriguing the grafting results, although they are not at this because of probably the blossom end rot didn't work much this year. But when you graft the hypersensitive variety from Esther on the tolerant eel, you get a clear effect of reduction of blossom end rot.
Yeah.
So that means that there's something that can act at the level of the rootstock.
Yeah, there's something. Yeah, something to work on. Yeah.
And for rootstock, you don't work with traditional rootstocks, which are also highly tolerant to various stresses.
No, we are using our material as rootstock.
Mhm.
Maybe could
Yeah.
be good to
Yeah.
have a control with
Yeah, with another.
one
Yeah,
of
yeah,
the
yeah.
best.
With one which is already known as
Maybe, yeah.
Okay.
Yeah. Good idea. Yeah.
Yes, this is Maria Jose. I'm working with rootstocks, and we use two commercial rootstocks as control, Beaufort and Maxifort, to test reels in cooperation as reference with the reels. And well, these commercial rootstocks are hybrid, so they are expected to be better than anything else, but not for everything, because sometimes for quality traits are not good under stress in comparison to reels. But in any case, as a strategy with rootstock, we are using or we are trying to combine both features of Drucker stress and salt tolerance to combine both. So I don't know if I did. And regarding your previous reports or papers on salt and drought tolerance with different locations, we have used, they have been very useful to elaborate the catalog, the QTL catalog. So we have used your data. They are already there. Thank you.
Maria Jose, because you produced this catalog, are you thinking about publication of this review of the information? Because and I don't know if it is a conflict with the deliverable being public, going public.
If there is a conflict.
No, the conflict can be stopped from being a conflict, because we can ask the officer to keep it within the consortium until it gets published. So we can act now before it's completely open, before it gets to.
Yeah, you are right. I think we have, as Tony has shown, there's quite it's quite interesting, these hotspots of QTLs for different abiotic stresses, and I think this review affords or it's interesting to be published. So I think
So do you want me to contact the officer and to change the status of the deliverable, moving it from public to public in the future, and now to be restricted to the consortium as deliverable? Because otherwise it will go open and then everybody can read it.
Yes, and it can be a problem also if you publish those data. Well, if you write the deliverable and then want to publish it, because it could be considered as plagiarism.
Yeah. That's why I'm
Yes, we have to be careful about that, because it happened to my colleagues that they wrote a deliverable with many details, and then when they wrote the paper, the materials and methods was the same, and so it was considered as plagiarism.
Yeah. For the future,
we have to be very careful with that, I think, Tony, because now as it is considered as public, also deliverable, we may maybe discuss with people in at the commission how to keep those data not public as long as they are not published. Yeah.
Yes, that's what I'm gonna do. I mean, the best is to ask the officer to change the status and to retract it. I mean, we can take it out of this public repository while we convince the publisher to publish the journal. Okay, because this will take some time.
And another remark for the catalog, maybe it could be interesting to do meta-analysis. There is a software, Biomercator, which is very easy to use and which will combine all the QTL data and map precisely how many QTLs in the region and where they are. So maybe it could complete the catalog.
Well, it's so beauty. The results are so beauty that even without any additional tool, you can.
Well, it's just statistics, adding statistics on the beauty of the results.
Yeah, we take the advice. Thank you.
Thank you, Mathilde.
Thank you, Mathilde.
Thank you very much.
So now, Mathilde, that you are just speaking, that you can continue
Yes,
for
I will.
conversation with us by describing your work package 7 on fruit quality. So, Mathilde, you are just on time. 90.
But I tried to share my screen, but I don't see it. Blah, 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 blah. Or phone. Maybe, did you stop sharing? I will. Well, I don't.
You don't see the green button on the bottom?
Yes, I see the button, but I don't see my, I have to authorize, allow Zoom to share.
Mathilde, do you want me to?
Yes, I think it will be easier if you can.
Okay, I will launch it.
Present. This is. You are the safety
okay.
person.
Just few seconds, and it will be online.
I'm sorry.
It's coming. Okay, you can go.
Yes, so the work package 7 is about improving food quality. And to prepare this meeting, we had a meeting with all the partners last month, where we reviewed all the process. So on the next slide, you will have the partners. Well, no, you don't have the partners. You have the complexity about food quality. As the other traits, it is a lot of traits. As the other work packages, it is a lot of traits considering sensory quality and nutritional quality, but we will go through later on. So next slide. You have all the partners who are working in work package 7, CC, KINRA, NEA, UPV, UNINA, and Gauthier, Semence, Villemorin, Orly-Magrin, Semi-Ortho, FCCB, and CTIX. So next slide. So the main objectives are to develop pre-breeding materials with improved consumer-driven traits for quality, flavor, and health-related. And there are 4 objectives. The first is to identify the most interesting resources for breeding food quality. The second, to improve elite material for specific targets related to flavor traits. The third is to assess the stability of the improvement in different environments or production conditions. And the fourth is to simultaneously improve the fruit's health-related characteristics and the shelf life. And so there are 5 tasks, and I will review the progress on each of them. So next slide. So the first task is the synthesis of resources available for improving fruit quality traits and identification of the most interesting characteristics for pre-breeding. As like in the previous work package, there has been many QTL and associations work for fruit quality traits. And so we did a synthesis of this data on the same reference genome and identify the most relevant QTL to be introgressed in the next task. And so this will, we did the synthesis to identify for each stress the most favorable QTL region, alleles, markers, and genetic resources. And the most significant one were chosen during the first 2 months of the project, mostly based on previous results from the project partners to be introgressed into different elite materials in the next tasks. And additional QTLs will be continuously monitored using standardized SOPs, and which will be published in deliverable 7.2. And the information should be uploaded in the Harnestom information site for deliverable 7.1. So next slide. So for the synthesis of QTL, we list the information gathered per each partner. And there are many different genes and QTL which have been cloned, which are in the literature, but also some results about some populations. So for INRAE, we have the QTL in the Serville by Leverville recombinant in red lines. And we have 3 publications about GWAS. The first one in 2014, the second in 2017, and the last one was in the frame of this project just recently published. In CESIC, there are also some data from reels combined with derived from crosses with Pampinellifolium. Also, the GWAS published in 2017, and soon there will be the Traditom GWAS panel. From UNINA, they ask if they have some relationship between transcript, volatiles, and sensory traits, and we were wondering whether it will fit in the database or not. And from NAR, they are gathering the G2P-Sol GWAS panel also. So the question was the availability of the templates and to know which data finally are needed for the databases. But maybe as we saw yesterday, those templates are almost finished. So maybe we have just to interact with Clara and Simo to finalize those data. Next slide. So, well, this is the same thing, and the questions that we had, we decided for the QTLs to work only if there is a physical location, and only identify those QTLs with small confidence intervals, because the other one may not be so interesting. And the same for the GWAS. And also we wanted to know what kind of data was needed for the database, because when you go from one study to the other, maybe it may not be the same information which will be available, particularly in terms of positive allele and haplotypes, which are not always indicated. And there are several candidate genes. Do we introduce them in the database or not? So maybe we have to discuss with this point. And we need to list all the information per partner, particularly for INRA, CESIC, and UNINA. Next slide. So for UNINA, they have identified from transcriptome data combined with sensory evaluation on the material from semi-ortho. They identified a list of candidate genes with differential expression, which could be interesting. And the question is how to take into account those data. Should we introduce them into TrumExpress? I don't know if it is RNA-seq data, but maybe Maria can tell us. And how to also to combine those data and their relation with the phenotypes. If we do it, we have also some data from Traditome where there was some sheeps on which we did some gene expression in relation to sensory traits, and which could be interesting to introduce into the database. So on the next slide. The task, next task is improving sweetness, acidity, and volatile composition through marker-assisted backcrosses in different elite backgrounds. And each partner identified his own genetic elite background and the QTLs which were the most interesting to introgress, and started to do it. So on the next slide, we will see per partner which QTL and which elite line. So for CIS-seq, they selected some lines, 6 introgression lines derived from Moneymaker by Pampi, carrying QTLs for sugars or volatiles were identified, and they are characterized here. And the work which is finished is already, they derived 200 F2 plants per IL, selected and genotyped for pairs of marker flanking the QTLs. They selected 70 F2 plants as recombinant and characterized them, grown them in FCCB greenhouse this spring. And they made crosses between introgression line selected to have some QTL combinations. And from the 70 F2 populations, they were genotyped by high resolution melting technique for 1 marker per 10 centimorgan to study the recombination in the QTL region. And then 5 tomato fruits were harvested per plant from trusses 2 to 4, frozen, and used for volatile analysis and bricks. On the next slide, there is the next steps. So they performed the sensory studies as shown on the picture, and with 32 untrained panels, we tested the tomatoes of 5 selected introgression lines with a triangular test. And they identified some differences between the introgression lines and the control. And particularly, 2 lines were tastier than Moneymaker. 1 with high bricks was sweeter than Moneymaker, and 1 with fatty acid volatiles were less tasty than Moneymaker. Then seeds from the F2 plants were collected for QTL, for further QTL validation, and they multiplied the population and keep leaf tissue. So the next step will be phenotyping for the volatiles, which has maybe started now, marker saturation within the QTL region to identify recombinants, and QTL fine mapping in 6 populations. And finally, DNA extraction and the whole introgression line collection for high density genotyping. Next slide. For Gautier Semence, they decided to introgress into their own elite materials some volatiles, improving sweetness without a negative trade-off on yield. And these are phenylacetaldehyde, phenylethanol, etc. But also acid content to lead to different sugar to acid ratios, and to introgress volatiles with favorable or unfavorable volatiles. At the end of 2020, they choose their elite materials as cherry tomatoes to introgress, and they produce the first F1. They choose the QTLs from previous studies, and they started in 2021 the first 2 backcrosses and made marker-assisted selection, which is ongoing, and there is no delay in the schedule. Next slide, please. In Limagrain, also, they decided to introgress into their elite material, aroma type, and the beef tomato hybrids. And the volatile compound selected are geranyl acetone, methyl cis-MHON, and all these volatiles. In 2020, they selected the hybrids for conversion, and in 21, they started to cross with sources that we provided from the core collection to create the introgressions. And they continue the marker-assisted backcross, and the work is on track. Next. In Semi-Ortho and UNINA, they made a compilation of all the literature, which is provided here with the different volatiles, which are supposed to be interesting for food quality, because often we characterize many, we quantify many different volatiles, but they are not all as important for food quality. And they characterize the volatile profile of 10 elite materials from Semi-Ortho, and the most promising hybrids will be obtained, and to do some F2 population to analyze at genomic level. On the next. Yes, this is some details. So this is a list of the material which have been selected. So mainly indeterminate, and some are long, pink, round, or long, red, yellow, etc. And the volatile analysis. And next. Well, the next task is from INRAE. We also choose one recurrent parent, which is on elite lines that we developed in INRAE. And we choose 3 donor lines based on their profile for volatile and sugar acids. And we selected some of these QTLs and started backcrossing, making marker-assisted selection and the second backcross. And we will continue next year. And there is no specific delay. So on the next slide, task 7.3 is the improvement of tomato health attributes and shelf life, as actually tomato genotypes with improved content of health-related antioxidant compounds, carotenoid or flavonoid, should be selected from previous phenotyping efforts and to be crossed with elite materials and look for segregation and identify the most interesting alleles to be introgressed. So at this first year, ENEA mined the Traditom and G2P-Sol data for finding some genotypes with altered antioxidant content and shelf life, and significant genetic associations. And in INRA, we started to prepare an F2 population derived from an F1 hybrid, which was created by our breeder in the past, and with a high food quality, which was shown in the Traditom project, and which is also combining high vitamin C and lycopene and also shelf life. And we will assess the QTL in this progeny. Next slide. So in ENEA, they identified in Traditom non-polar compounds and reanalyzed the data. And semipolar compounds also, the data are analyzed for correlations and GWAS. And firmness data are analyzed and correlated with metabolomics. And so they will get, at the end of the year, the elite genotypes with improved semipolar content and firmness. From G2P Solves, the SNP data are available, the GWAS is underway, and they have data of non-polar and semipolar compound available, and the list of elite genotypes with improved antioxidant content and firmness will be available at the end of the year. Next. So the analysis have to be completed, and well, this is the same, I think. Well, yes, and for Ernestam, they will prepare some crosses between elite genotypes and decide which parental lines. And we decided that one F2 population will be characterized in 2023, and it will be field grown in INRA and phenotyped in NAR. Next, and as I said previously, for INRA, we have prepared an F2 population from our hybrid Garance, and next year we will phenotype and genotype it to assess the QTLs. And one of the parent, which is already resequenced, but the other one will be resequenced in the work package 4 to have the whole genome of both parents. The task 7.4 is the optimization of pre-breeding for several quality traits simultaneously. As you know that quality is not only one trait, and so we will optimize a recurrent selection strategy by genomic prediction of multi-trait prediction, and perform some the most appropriate crosses to obtain the next generation. And in the next slide, we decided to start preparing a multi-parent MAGIC population, but with selection to increase the frequency of positive alleles for quality traits. So this year we chose the parents and started doing some crosses and optimization of the design. Next. And we will prepare a MAGIC pop with 2 large fruit lines and 6 cherry tomato lines, which are providing some complementary alleles for the volatile QTLs, and then make a combination of those lines and those F1, and then do intercrosses and selection in order to have the possibility to genotype and phenotype the population before the end of the project. And this will be performed in collaboration with Villemorin and Limagrin. In semi-ortho, in collaboration with UNINA, they will do the crosses, also appropriate crosses by genomic prediction to obtain new generations. Next. And so in the task 7.5, which has not yet started, we will study the stability and robustness in different conditions. So first, we have to list the protocols for sensory assessment and consumer tests in deliverable 7.2. And then the original materials and the introgressed one will be assessed in different cultivation conditions across Europe and beyond. And this will be performed by several partners, but this, as I told, didn't start yet. Next. So to be done later in this task, semi-ortho will evaluate the selected material in different conditions for sensory evaluation and using a trained panel. In CSIC and CTAX, They will also participate in the evaluation, including metabolite profiling of advanced material. In UPV and FMA, they will evaluate the material and excuse me, and sensory evaluation of 12 selected varieties will be performed using a trained panel. So this is the list of the deliverables, and we can see that there are none on the first year, but we will have to finalize a report of QTL alleles and genes to be introgressed, and the SOAP also for next year. Next. The activities planned for next year, well, enter the data in the database in relation with work package 3, start listing the SOAPs, and for the task 2, we will continue the introgressions. Task 3, we will make the crosses and QTL mapping. In task 4, they will continue the population development, and task 5, maybe we should start listing the materials or type of materials that we will be studied, and the timetable for sensory tests. And dissemination, we published one paper about, together with CSIC and Gauthier Semence, about the QTLs in tomato hybrids. And there was also a dissemination in a newspaper talking about the work in Harnesdam and the lines which are studied by UNINA. No main deviations presently. And for the milestones, there was a milestones due on month 12, but for the design for marker-assisted breeding of fruit quality, but there is no report on this. And each marker-assisted backcross program started yet. So, is there any question? And maybe some compliments by the other partners, because I just summarized their work.
Just wanted to make a comment, Mathilde, is that the sensory analysis that we perform in our introgression lines, initial materials, to confirm that the differences that in volatiles that was the basis for selecting those, the panel was able to differentiate. This was done by Fundación Miguel Agustín, not by ourselves. So this was just to make it clear that we didn't give the appropriate credit for that.
Yeah, sorry.
No. And I think it would be good also to know the QTLs that Gauthier is working with, because I saw already Bill Moran is working with some of phenylacetaldehyde in chromosome 9 that we are also introgressing. It would be nice also to put together all this information from CSIC, you and Bill Moran and Gauthier to see how many loci or regions that we were talking about and possibilities also of interaction. Yeah? Yes,
of course. There are several for phenylacetaldehyde, and they will be introgressed in different elite materials. So it will be interesting to follow the final materials and compare them. Yeah.
Because so far here at CSIC, we are just doing the fine mapping and using the initial yield population, yield lines selected. It would be nice also if it's useful to do the crosses to the elite materials in either of these two companies as partners, if they want. Yes. For UNINA, I think it would be also good because for the review of some of the papers, some of these papers that you show in the table were also reviewed by some of us, by Mathilde, by Jordi here at the CSIC. It would be good to coordinate so that we don't duplicate efforts, right? And also you indicated, Mara, that you will be doing volatile profiling using PTR TOF. And our experience with this technology, it didn't work as good. So only just for you to know. For us and for Arnaud Bobby, it's good for processing lots of samples, and it's fast, but I don't know, the quality of the data that is in our hands or the information is what was not confirming what we have when we do the standard ECMS on Headspace or using SPM. Right? So that's
okay.
Just for you to know.
For that, at the moment, we have not yet the result they got from the machine, so we don't know.
Okay.
We are waiting.
And Mathilde, I'm pretty sure you're aware of this, the ethics requirement for the taste panel and everything. So the consent of we need to make sure that we comply with the regulation. Otherwise, as you know, publication, and we don't want to go in trouble with the EU, but it's only.
Yes, maybe we have to tell that to all the partners. If we want to publish at the end of the project some sensor, well, or at any time, some sensory data, now it is mandatory to obtain some certificate of from each panelist that they agree with the test, and to have an ethic. Maybe we could look at a publication of sensory traits. What are the mandatory certificates? Because we had the problem previously with Tony and Silvana. Yeah. Later, it is really difficult to obtain all the signatures and certificates. So now we know that it is mandatory, and it has changed recently, more or less recently.
We did with our test. We asked for signature. For our test, we have this signature form.
Any other question for in relation with what package 7?
Yes, may
Andrea?
you hear me?
Better if you speak louder.
Okay. I would like to ask Mathilde, which is your feeling about doing a sensory test with untrained people? I mean, which is the trick to get the best objective results from them? Do you have any?
You're muted, Mathilde. Mathilde, you're muted.
Sorry. I think that for triangular test, it is less, well, it may be okay. Usually it is better to have trained panelists, unless you want some consumer test, but you need a lot of consumers. For sensory profiles, it is really better to have trained them. Maybe Clémence, you can talk about your experience in Gauthier Semence, but you train the panelists.
Yes, hello. And also my colleague Pauline Mouton, which is here. She has the more background on the internal sensory panel that we use here at Gauthier. We have an internally trained panel, and yeah, we really see difference through the training session, especially in the capacity to decipher. Are we testing the same sample, just some repetitions, or different samples? And also with some bias. If we detect, if the consumer is not trained, there is a tendency to associate high sugar with good taste and not to decipher between aroma and sugar. So it depends on the objective. If it's just a general sugar and acid traits, I think there will be less bias with untrained panel. But if we really want to study specific volatiles, we really need to have a train, and also we try to train panel really volatile specifics and not just with some taste family like fruity or green and so on. And yet then we really have to develop kind of protocol for each volatile if we want to do this kind of work.
Each aroma, yeah.
Yeah.
I think, Andrea, the best is to ask the people with expertise, but in the rule of thumb here is that considering that each of us are different and we differ in 3 levels of magnitude, our sensitivity to different volatiles, and there are 1,000 receptors for volatiles in our genome, and considering there are 100 volatiles or maybe 20 plus volatiles involved in flavor, then the more people you have, the better. And if to describe things, you need trained people, because lay people just don't know how to describe what they feel. So if the question is this variety more appreciated by consumers at large, then you need a large panel of consumers and trained. But if you want to have answers to describe better your material, then you need trained panels, and this takes time and money and effort.
And according to the descriptor, some of the traits like sugars and acids are more easy for people to characterize, but you can also characterize just that by looking at the sugar content and the acidity. But for volatiles or texture, you really need to have some trained panels. And it also depends if you want to characterize some materials for a breeding company, just look if they are really bad or really good, or if you want to publish. If you want to publish, then you will have to precise how the panel was trained and how many people and how did you design the training panel. And when you have a small set of varieties, it is easy to design the protocol. But if you have, for instance, QTLs, if you want to map some QTLs for sensory traits, then you have to be very careful in designing the protocol and the repartition of varieties per trained, per panelist. And that's what we did with recombinant inbred lines when we did some QTLs for sensory traits. We added at each time the same F1 hybrid to have a control for each panelist. And we saw that we could characterize the quality of the panelist with that.
Okay, because the design is incomplete in that case.
Yes, because you may test maybe 6 to 7 tomato fruit, no more. Then you are saturated and you are no more efficient. And the first one is always more sweet than the last one, whatever the sweetness.
That's true.
Thank
and
you.
Of course, I'm sorry to confirm that I still have not recovered my sense of smell since I got COVID last year, Christmas Eve. So I don't know.
Maybe now it's
all these
criteria.
All these approaches about improving flavor will change with more people like me losing the sense of smell. Hopefully not.
So maybe you have to select your panelist from the absence of COVID.
And you can require antigenic tests to
Yes.
But you can still probably sense sugar, right?
Yes, sweetness, sugar. I can.
It is with the tongue, so
Yes.
yeah.
Okay. Is any other question or comment or issues to discuss?
I want just ask to Simo, actually, not Mathilde, for the question on transcriptomic data and metabolomic data. If there are interests from our platform, if could be of interest, upload them. The one that we have related to this.
Are they RNA-seq data or
Yeah.
just, yeah?
Yes, for definitely for the Tom Express, yes, it will enrich the database. If we have this kind of data, we can of sure we will include them. Yes.
But what is maybe more difficult is to relate the presence of on the same material of sensory traits and.
For the moment, we didn't think how to have a tool that can analyze at the same time the phenotype and with the RNA-seq, but maybe it will come later. But at least to have this data in the platform, this will be good.
Okay.
For people that want maybe to do the analysis by their own tool, so they in the same platform that they can download the data and they do the analysis. So at least to have them in the Harness-Tom gateway, this will be good.
Okay, so we'll contact you to know how to send you.
Okay.
And for the database, maybe could you tell us when, or Clara, when do you think the forms for the QTL to enter the QTLs will be finalized?
Okay.
That we start preparing those data files, because we started with one form, but I don't know if it is the final one. And we had several questions and we started discussing with Eden and then we stopped. So we have to start again.
We have also in our team, David Pierre, that is here also, that he,
yes, and David
is working hard to finalize the templates, and as soon as it will be ready, we will circulate them to partners, and we will of course send it to you, Mathilde.
Yes, and so maybe with Mara and Tony's, we could discuss about valorization of all this data. Yeah.
Okay.
Thank you. So we have a coffee break now, and I wanted to ask you something because the policy officer expressed the interest to join today's meeting, specifically for Work Package 8, that she has a special interest, and this is scheduled to start at 10:30. I would like to ask you to come back to the meeting at 10:30. I know it's like 20 minutes longer than, because this is according to the schedule, and we are now 20 minutes ahead of the schedule. But this is just to be polite with the officer in case that she connects. It's the policy officers, so we need to be nice with her. Okay, so what about reconvening here at 10:30? Okay, for the Work Package 8 presentation and discussion. It sounds good.
Okay.
Are you okay with this?
Yes, okay.
Okay.
Okay, fine.
Thank you. Thank you. I'll see you later. Bye. Aure, sube aquí.
Come here, my friend.
Come here.
Oye, el Aure se ha puesto barba ahí de profeta, ¿eh?
Como Heidi, macho.
Joder. Bueno, entonces a las 10:30, ¿no? Hasta luego.
Ahora.
segundo.
Está en el...
Vale. Porque así ya no tengo que buscarlo para buscar.
Ahí hay un enchufe.
Sí. Una pregunta, ¿los workshops de qué son? ¿Los workshops?
No, en principio no hay de
No girando esto, es que cuando yo giro la pantalla se desenchufa. Y entonces me ha dicho Miguel, ten cuidado porque es que si no está muy ajustado te va a quedar sin...
Está puesta la cámara.
Sí, la cámara sí, pero no me importa.
Okay, welcome back to the to the session of the annual meeting. Hope you enjoyed a good coffee. We are back for presentation and discussion of the Work Package 8, which is the participatory breeding and citizen science type of more complex title, but it goes just to summarize the context. It will be Maria Jose from the University Polytechnic of Valencia to present it. And I think it's just time to start. Maria Jose, the floor is yours.
Okay, thank you, Tony. I am going to share my screen. Can you see my screen?
Yes.
Yes?
Yes.
One moment, please. Okay, now it's okay? Yes. Good morning, everybody. I'm going to present activities that we have conducted in this work package. Work package 8 focus on participatory plan briefing and citizen science, connecting genetic resources, plan briefing, and scientific methodology with society. The work package is led by Alcalax and UPV, and the presentation, of course, has been prepared with the contribution of all partners. We have 10 partners in this work package, with a total of person months on 173. And there are 6 partners that contribute with more person months, okay? The general aim of this work package is to involve farmers, consumers, chefs, and academy in the pre-breeding and breeding activities. For this purpose, we will design innovative strategies on participatory plan breeding. This general objective will be achieved by these 4 partial objectives. The first one is to develop innovative strategies for involving other actors that are part of academy and breeders in pre-breeding and breeding activities. The second one is to design ideotypes regarding agronomic performance and sensory quality based on breeding materials and local varieties. The third one is to take profit of the work done in previous EU-funded projects by selecting the most promising materials and developing new materials from them. And the fourth one is to establish efficient ways for transferring germ plants to farmers and citizens. About the significant findings achieved until now, I would like to highlight the ones related with the first 2 tasks of the project, task 8.1 and 8.2. Task 8.1 deals with the definition of ideotypes by farmers, consumers, and chefs. In this sense, we have already designed specific surveys for drawing agronomic and sensory ideotype based on gastronomic and cultural tradition of each area. This survey will be presented when we talk about task A1. Related with task A2, which is the constitution of a repository of genotypes previously identified or developed as promising in other EU-funded projects, we have selected a set of 241 genotypes, including landraces and elite genotypes. We will explain in some detail the process we have followed for the selection of this set of genotypes. Going now to the process towards the different tasks, we will start with the task 1 that will be presented by our colleague, Joan Casals, which is the main contributor to this task. Of course, other contributors, all the partners of this task, of this work package have contributed, but Joan has been the main contributor. So he's presenting the activities that he has done. Joan, please, are you ready?
Yeah, thanks, Maria Jose. So good morning to everyone. I am Joan Casals from Polytechnic University of Catalonia and Fundació Miquel Agustí. So as you can see, the main objective of this task 8.1 is to integrate the preferences from farmers, consumers, and chefs, and to draw different ideotypes. To do this, our starting point was the previous published works, where it was described that there is a consumer segmentation regarding the sensory ideotypes, and also more recent works where it has been compared the sensory profile of tomato land races and modern varieties regarding the consumer preferences. So our objective is to combine the viewpoints of farmers, chefs, and consumers, and to analyze if there are regional differences regarding the tomato ideotypes. And also we want to address and to try to draw specific ideotypes for some study cases, some land races that partners are interested on. So next slide, please, Maria Jose. So to do this, we started organizing a focus group with the participation of farmers, chefs, and consumers. And this enabled us to list the most important agronomic, commercial, sensory, morphological, culinary attributes that we should focus on, so that we should study in the different the survey that we have designed. Next slide, please. So we have implemented an online survey in an online platform. You can see there the structure of this survey. For sure, there is a common part at the beginning regarding the socioeconomic profile of the participants. And then the survey is split in different parts regarding the different study cases that we have in this survey. So you can see there that we are trying to make the ideotypes for the Valenciano, Oxheart, San Marzano, or Montserrat tomato land races, but also people that do not know these varieties, they can help us in drawing the ideotype from a general tomato. Then you can see also that there are specific questions for consumers, chefs, and farmers. So at the right of this screen, you can see some drawings that have been produced specifically for this survey. This was done in order to facilitate the interpretation of the traits that we are asking about in the survey. And you can see, for example, different intensity of green shoulder, different fruit colors, or different fruit ripening. Next slide, please, Maria Jose. This was before, okay. So there the list of questions that we are asking to the people participating in the survey. I will not go in deep in this, just to signal that, as you can see, we have many questions for farmers. Next slide, please. So the survey has been translated in a different language. Now this survey is available in English, Catalan, Spanish, Bulgarian, Italian, Dutch, and Chinese. So there is no excuse for not participating in the survey. And I really will like to thank to the different partners that have participated in the translation, because I know that this was a huge effort. Next slide, please. So there you can see some image of the survey, but I think it's more better if you perform the survey and you can check it by yourself. Next slide. So just to finish, this survey will be published next week, and we expect that it will be open till beginning of February. We have made some tests with students that we have, and you can see there that we have already more or less 600 answers. Our objective is to have at least 2,000 answers, and we have a more ambitious objective is to have 2,000 answers per each region where the survey is disseminated. Let's see if we achieve this. In any case, we will need your help in the dissemination process. And I think that next week we will send to all of you, an email with some instructions on how to make this dissemination. So that's all for this task 8.1. Thanks for your attention, and please help us in the dissemination process.
Thank you, Joan. I follow with the second task, the A2 task, which is the constitution of a repository of genotypes previously identified or developed as promising in EU-funded projects. The idea of this task is to take profit of the EU work performed in several EU projects on tomato by selecting a subset of materials interesting for several characteristics, for their good agronomic performance, because they are well-known and appreciated traditional varieties, genotypes with resistant to diseases or tolerant to abiotic stresses, etc. So we started with by doing some discussion between several partners and general discussions also among all partners of this work package. As a result of this discussion, we agree on some selection criteria that I will explain later. Then we apply this selection criteria to each project, and the project with which we worked were Traditom, Brezov, G2Psol, Tongem, and Route to Power. The selection of each project was made by this institution, our colleague of FMA, UPV, Maritza Institute in Bulgaria, UNINA, and IBIA. After this first selection by all the partners of the work package, we check duplicates and underrepresented diversity. And the last step was doing a joint analysis to see what we have selected and what we have in this selection performed. The starting materials were those that we have here in this slide. We started from almost or a little more than 3,000 accessions. And you can see here the number of accessions of each project. For all projects, we have a lot of data. We have, for Traditom, Brezov, and G2Psol, we have breeder selection. And this was a very valuable information because was the breeder selection made directly for breeders in the field of the experiments. So we had also phenotypic data for these three projects, phenotypic data. In case of Brezov and G2Psol, we have also data about resistance to diseases. And also we have genotyping data. But the problem is that the genotyping was performed with different technology in each project, GBS, DBRAD, SPED technology. In case of Tongem project, we have data about heat tolerance. And in case of Route to Power projects, we have information about the drills conducted from the cross between A. thraciforme, susceptible to salinity and sensitive to salinity, and a section of Pimpinellifolium tolerant to salinity. So this was the starting material. So the criteria agree, we agree of 5 criterion. The first one was the selection of the genotypes with good agronomic performance and good quality fruit and good fruit quality. For this criterion was very useful, the breeder selection conducted by breeders. The second one was the selection based on phenotypic data, growth type, yield, fruit weight, Brix degrees, just to have a good variation in the repository that we were selecting. The third one was the selection of the best accessions of the most relevant European traditional varieties. In case of Traditom, G2Psol, and Brezov projects, we had a lot of European, well-known European traditional varieties. So we try to have a good representation of the most relevant, of the best accession of these traditional varieties. The fourth criterion was the selection of accession with resistance to diseases and heat tolerance, regardless of the agronomic value of the accession. And the last one was the selection of the best 2 or 3 reels to be used as rootstock. So the final number of accession selected for the repository was 241, and you can see the number of accessions selected from each project. Here you have some representation of some important agronomic traits that we have, the diversity that we have in the repository for these traits. For example, for fruit weight, we have a good variation from very small fruits to until 700 grams. For the soluble solid content, which is very important, quality trait, we have also a good variation You have here the world repository, and here split into the different EU projects. We have a good representation also about varietal types. Most part are fresh market, but we have also long cell life, processing varieties, cherry, cocktail, and we have also variation for the growth habit. For other traits related to fruits, as ribbing, fruit shape, good variation of all shapes, fruit color, green shoulders, which is very important in terms of the preferences of different markers. Puffiness, normally puffiness is a non-good characteristics of the fruit, but there are specific traditional varieties in which this characteristics is very valuable. For example, the Montserrat traditional variety. Firmness. And here I want to show you some of the accessions selected. This one from the project Traditom, the world collection of Traditom. These were selected from the core collection of Bressot project. You can see the variation. You can see also the good agronomic performance in terms of set of fruit and characteristics of the inflorescence. And here you can see the selections made by breeders, both in UPV and Maritsa Institute in Bulgaria, in the project G2P Sol. We have also around 40 accessions with resistance to several diseases, viruses, fungus, and bacteria. And I have to add that yesterday was said by Gancho, our colleague of Maritsa, that this resistance has not been confirmed. So we will have to modify this table. We have also about 30 accessions with heat tolerance selected in the TomGem project in Bulgaria, Italy, and Spain. And about the reels, we have in our colleagues of IBIA made a selection of the best reels in terms of fruit yield, solid content, and tolerance to salinity. And these 3 reels will be used at rootstocks. Now the following with the progress toward task. Task A3 starts on month 13, so we will deal with this task in a more advanced with the presentation, where we will talk about the progress for the next year. The task A4 is participatory plant breeding and evolution of land races. In this task, most part of the partners of this work package participate with different breeding programs. Most of them related with introgression of resistant genes into the elite varieties, traditional varieties. And I am going to give the word to each partner, just in 1 or 2 slides, explain what are they doing in these breeding programs. The first one is the breeding program on the Penchar tomato, which is carried out by our colleague of FMA, Joan. Joan, are you ready? Just to explain this slide, please.
Yes, Maria Jose. So the objective of this breeding program is to introduce resistance to tomato spotted wild virus, verticillium, tomato mosaic virus, and fusarium to a Penja line, the LC42. The starting point of this breeding program was the work done by Dani Zamir and people from their lab in the Traditom program, where they achieved the BC1 and BC2 generations. So we are following this backcross breeding program. We are now at the BC4 and BC3 generations. We are also obtaining some homozygous resistant lines by using just selfing. So we are also selecting some lines, and we are just advancing in this breeding program. In the image at the right of your screen, you can see the diversity in fruit shape and fruit color that we have seen this summer in the BC1 and BC2 generations. you can see that there is much diversity as expected in this in the first backcross generation, and in the second backcross generation, we are recovering very well the fruit shape of the original line.
Thank you, John. From Maritza Institute, they are performing two breeding programs. Vania, are you ready just to explain a couple of slides?
Yes, Sam, thank you, Maria Jose. The aim of our breeding program is to improve the Oxford landrace in order to become resistant to TMV. This landrace, Oxford tomato landrace, is very popular and widespread in Bulgaria. It is distinguished with very large pin-colored fruits. And we used our elite line 975 as a donor of resistance in a hybridization program. We did 2 back crosses before the project start. In the current season, we received BC3 generation, but also we made the self-pollination of BC2. And the plan for the next season is to make 1 more back cross up to BC4 and the self-pollinated seeds from BC2 and BC3. The crosses that we made is with TM22 heterozygous plants, which are proved molecularly by CAPS marker. And since this is quite advanced program and we will reach our target probably next year, we started a new program. If you go to the next slide, please.
Yes.
Yes, this season we started a new hybridization program with land races, which aim is to improve their productivity. And we made several crosses between pink-fruited genotypes and between red-fruited genotypes, and obtain F1 hybrids. And our plans for the next season is to evaluate F1s, but for agronomic and fruit quality traits, and to continue with back cross with some of them or with all of them, it depends. Yes, that were our activities and plans here in Bulgaria. Thank you.
Thank you, Vania. Now we have our colleagues of UNINA, which are conducted also two breeding programs. Amelie or Mara, can you explain this slide, please?
Will be a PhD of Amelie to explain it. Is Salvatore. Salvatore?
Good morning.
Okay,
can you
Salvatore.
hear me?
Okay. Yes.
Good morning to everyone. The first breeding program is focused on breeding of 4 San Marzano land races to genetically improve them for the resistance to tomato spotted wilt virus and verticino wilt disease. To achieve this goal, the 2 resistance genes were investigated in F4 genotypes from Dochet hybrid with the aids of 2 molecular markers. Among the several number of plants investigated, only the ones showing both resistance genes in homozygous condition were chosen as male parents to perform the crosses with the 4 San Marzano landrace. Now we obtained the F1 seed from the crosses, and we are using it in the backcross scheme. The second breeding program, please, thank you. The second breeding program allows to develop a novel genotype for tomato serbo market with the tolerance to high temperature, disease resistance, and good fruit quality trait. To achieve this goal, A42 genotype was chosen for the quality traits, the fruit quality traits, the yield stability under high temperatures, and the high tolerance to tomato spotted wilt virus. And this genotype was crossed with 5 indeterminate tomato accession. We obtained the F1 seed now, and we are starting the backcross scheme.
Okay, thank you, Salvatore. In UPV, together with Alcalax, we are conducting also 2 breeding programs. One of them is for introgressing resistance to tomato spotted white virus, tomato yellow leaf curl virus, Fusarium, Verticillium, and nematodes in 3 Depenchard tomato lines. So we started by crossing these 3 Depenchard lines with 2 donors of the genes of resistance to these diseases. The objective is to have to join different genes of resistance, always having the gene of resistance TM22, which is very important for confer resistance to the tomato mosaic virus. We performed the first crosses, the BC1, and at present we are performing the BC2 generation. The other breeding program is also in Depenchard tomato, and we is the development of new Depenchard tomato varieties with high agronomic value and resistant genes. For this purpose, we have crossed the same 3 elite Depenchard lines with 12 Depenchard local varieties selected for their agronomic values. And you can see the standing characteristic of these 12 varieties. So we have cultivated the 36 hybrids and the parentals in an assay. And currently we are analyzing the data and make selection of the best hybrids to be checked again in the next season. And the last program is Perforban Unitus. Andrea, can you comment this slide, please?
Yes, sure. Good morning to everyone. Do you hear me?
Yes.
Okay. So this task is aimed to prepare a multi-parental population to be used in participative selection schemes using local varieties, landraces from the region of Latium, which is the region where we are located, and from the region of Tuscany. All these varieties are within the flattened ribbed typology of tomato, and the 4 landraces will be crossed, well, have been crossed because this has been done this year with the tomatoes with the red frame, which are commercial hybrids obtained in the same typology or more or less in the same typology. So we are now growing the first F1 hybrid to do the second cross, and we will do the third cross in the 2022, and we will have the multi-parental population done by the end of next year. We don't know if we can actually do the participative selection within the Harneston project, because this was not planned at the beginning, but this population is very some regional agencies are very interested in this kind of population, so we hope it will be useful for the next activities in next projects. And that's it for this.
Okay.
Yeah.
Thank you, Andrea. Following in the task, we arrive at task 8.5, which is participatory plant breeding to diversify tomato color and quality. The responsible for this task is Andrea also. And Andrea, please, can you explain this task?
Yes, this is a participative scheme built to select within a population characterized by yellow color of the fruit or orange color of the fruit. As you see, the population has been done previously. In this case, they were already done, so we could start with the selection this year, and they come from crosses from different types, so they do represent quite a lot of variability. As you see, they have been grown in 2 different farms, and we conducted this experiment in parallel in Italy and in Spain. And in Spain, it was the colleague Joan Casals to take care of these experiments. And in Italy, we got the help of a subcontractor, which was Rete Semirurali, rural seed network, which has quite a lot of experience in participative plant breeding experiments. So the idea is to select the best 20 plants following the judgment of farmers. Next one, please. And this slide show you the places where the fields have been done in Italy on the left and in Spain on the right. And some shots of the participative fields. And in the middle, there is our advertisement for the event of the selection. We ourselves took a number of traits. We phenotyped ourselves the plants during the growing season, but the selection was done in a single day with farmers. I can say we couldn't reach many farmers this year. The selection has been done with 7 or 8 farmers for each field. Maybe this was a problem because we were a little bit late in the season and the selection was done in mid-August, which is not actually a good period for many reasons. So we will try to be earlier next year and to have more farmers to work with us. So in the next one, just a few results. We are, okay. No, never mind.
Excuse me.
Don't worry. No. So yes, in the first one, we are elaborating the data. So we have preliminary graphs. This is representing the earliness of plants in different populations. The two populations selected are actually the second and the third couple of graphs. And as you see, Italy is the blue one and Spain is the yellow one. So as you see, the environment was very different because all populations flowered earlier in Spain. And in the next graph, you see one of the traits that we took. And this is about inflorescence type. If you look at the right of the graph, you see the Italian populations, and the second mosaic shows the whole population. The second one in the second column, and the first column shows the selected population. So you can see the switch in inflorescence type that has been gained with the selection. And the idea is that the farmers did select better plants with uniparous or with fishbone inflorescences. And the next panel, you see some results of the score that have been done by the farmers. The farmers had a very, technically simple task that was to judge the plants with a score from 1 to 4. And this is a regression studies of the number of fruits against the farmer score. And it is interesting to note that we have a country by color interaction, because in Italy, in the first column, you see that there is a positive relationship between the number of fruits and the score for the orange population on the top, but not for the yellow one, while exactly the same, the contrary, sorry, happens in Spain. So we believe that studying this kind of correlations would give us quite good insights about the idiotype that the farmers see, and that it could be a good advice for next breeding activities. So this is what we did, and a lot of data could can be elaborated in the next days and will be presented in the next report.
Thank you. Andrea. So we have in our work package several deliverables, but 2 of them are due to month 12, and we presented without problem. So we are okay with about the deliverables. About the planned activities for 6 and 12 months, regarding task 8.1, we have this planification, first to disseminate the survey, and it will be active from October to January 2022. Then we analyze the data in February, March. We will have defined the ideotypes by April, disseminate results, and integrate results with other work package tasks. About the task A2, which is the repository that we have selected from the previous EU projects, we will finish phenotypic characterization and seed regeneration if needed, and we'll genotype at low density the accessions we have included. About the task A4, participatory plant breeding and evolution of land races, I am not going to go in detail, of course, in these 8 breeding programs, because they have been already explained by each partner. And just to say that all of them we will follow by doing the following generation, F2, back crosses, and so on in each work program. Task A3 starts just now, and the responsible for this task is Andrea. So I give again the word to Andrea. Please, can you explain something about this, the planification of this task?
Okay, thank you again. Task 8.3 is related to the citizen science experiment and participatory plant breeding to improve the efficiency of tomato breeding. So in this task, we want to reach not only the farmers, as we explained before in other tasks, but also to reach the citizens, students, chefs, and other consumers of tomato with different roles on the choice of products. So the idea is to use what we call the participatory plant breeding kits. I will explain a little bit more in the next slide, and to do also a taste experiment in the second year of the task, and also to organize courses and seminars directed to consumers and to schools or chefs. In the next one, you see a little bit of detail on the citizen science and participatory breeding experiment that we want to do. Starting from the selection of varieties that has been done in task 8.2, and that Maria José has explained before. We selected 26 accessions to build what we call the plantlet kit. These accessions will be actually 25 plus a commercial control, and they will be distributed with 4 plants for accessions, so using some 100 plants to farmers in Bulgaria, Italy, and Spain, the countries that participate to this task. We plan to reach a minimum of 15 farmers per country, and we will follow the farmers to get their opinion, their selection, their preferences, and so on. On the other side, next panel, yes. We also plan to distribute these 25 plus 1 accessions to citizens in the form of seed kits, which means seed packs with 5 accessions, 1 control plus 4 of the selected one. And we plan to, of course, the citizens would be selected somehow for their motivation. They should be registered. We should ask for the informed consent, for the privacy procedures, and so on. Then we will send the seed together with the instructions that they will also find in tutorials in the website. The citizens are supposed to grow the plants to give some score on traits, and hopefully we would reach at least 260 citizens in such a way that each variety would be repeated about 40 times. But these are minimum numbers, so we hope to do more than this. One important thing in the next slide I can clarify this is to understand exactly have an idea before planning the experiment on what we want to give to the farmers and to the citizens. So I think it's important that the Arneson partners can try to transfer. Go ahead, please. Yes. They can transfer information and awareness about the importance of genetic resources, of diversity, plant diversity, in the meaning of different uses, varietal types, typologies of the fruit, shape, color, origin, and just to mention something. And also we would like to get the farmers and citizens to work on plant and fruit traits which are important in breeding, like growth habit, inflorescence type, and flowering date, and traits regarding the fruit, of course, such as the number of locules, the color of the fruit and the skin, and so on. On the other side, we would like to get back something from them, which is an idea on about their interests and preferences. And it would be interesting to understand how different people in different environments would make scores on the same genotypes. So we will have a G by E by O interaction. We will ask them to possibly send the pictures of what they got in the experiment, so that we can check a little bit if there was any mistake or any admixture of labels or plants. And this is in the final goal of getting new roads to breeding tomatoes. In the next slide, there are some thought about how to implement all of this. This has to be done in the next month, so we have to be fast. I wouldn't go into details in this, but just I would like to tell that the citizen part of the experiment would be more tricky. And for this, we get an interaction with the coordinators of the project Increase, which is doing a very similar work, maybe on a bigger scale, in bean diversity in Europe. Of course, we will ask the collaboration of all the colleagues involved in the task, and also of colleagues involved in other work packages, as we said yesterday, especially work package 2 and 3. Okay, I think this is the last for me.
Yes, this is the last for you, Andrea, because this is task for task 8.5, that you are also the coordinator of this task.
Yes,
and,
this is what we are going to plan the next year. It is quite simple. We want to continue the participative pedigree scheme on the yellow and orange varieties in Spain and Italy. We will correct some little problems that we had, like the late season of this year. The aim of this task is to get in year 4, fix the selected lines, pilot F1 hybrids between them, under the mixture of the selected lines, the original population, which we will maintain during the years, and the contours that we already used. So with this comparison, we could understand if this material, which material is better adapted to the farmer needings, especially in low input and organic conditions.
Okay, thank you, Andrea. So for dissemination and training activities, Work Package 8 had a meeting with Work Package 2 just to coordinate these activities in July 2021. And we established a calendar. Each week, one of the partners upload a dissemination activity. As we are several partners in this work package, each of us has to upload an activity each month and a half, more or less, which is not very much. And we are always more or less continuously updating some small activities. For example, I can give you some examples here. For example, in UPB, participate in a competition for the best tomato. We also participate in a talk given to the Association of Productors of Valenciano tomato in Valencia, in Meliana. And we made also an interview to the president of the Association of the Productors of the Penchar tomato in Alcalá de Xivert in Valencia. Other activities have been also conducted by our colleagues of FMA. For example, a seminar about the watering, how to use water for more efficient watering, and participatory plan bidding from journeys organized by FMA also. And our colleagues from Bulgaria, from Marisa Institute, presented the project in the roundtable Perspectives for the Development of Innovation in Bulgaria in October 2020, and also was presented the project in the European Corner during European Research Night. And our colleagues from UNITUS, Andrea and his colleagues, have also made participative selection of yellow and orange tomato segregant population in Italy, as he has already described. And he sent some news and some images to Work Package 2 for the dissemination. About problems and deviation, we have only a small deviation. The deliverable 8.3 was due for month 24, but this deliverable is related with the task 8.3, which finished in month 36. So we have delayed the presentation of this deliverable to month 36. This was already, Tony is already informed about this deviation, and we expect no problems with that. And thank you for your attention. This is all about the presentation of Work Package 8. If you have any question, my colleagues and me will be happy to answer.
Okay, this is Tony speaking. I'm checking if Annette is still connected. Annette, are you there? I cannot see you now. Okay, so our policy officer, Annette Sniggans, was connected until a couple of minutes ago. So probably she just joined us to attend the Work Package 8 presentation, but I don't see her now. Are you there, Annette, under a different name? Or just not anymore? Okay, so we can proceed with the discussion. Okay, if Annette joins us later, we give the word so she can tell us more about the perspective of the DG Agri about our project. So, anything for discussion for Maria Jose or any other of the participants? I have just a comment, because I think that the participatory breeding on the color orange and yellow makes a nice it's a nice trait to select, and the combination of shapes and colors is very easy, colorful, and variation is obvious. Is it possible to do something like this for taste? Could it be possible? Because it's what derives most of the desire on tomatoes about taste. Of course, color is important and shapes are important. But what do you think? Is this? I mean, this is what you can do more easy. But what about taste? Is it possible to do similar things with taste?
Yeah. Joan, you have a word more on taste than me. And Andrea, Joan, can you answer this question?
From my part, I agree with Tony. This is a really nice idea. The problem is that with the experiment that was designed by Andrea, we have worked this year in each F2 populations with maybe, I don't remember, maybe 150 or 200 plants, segregating plants. So yeah, I would like to ask to farmers to eat individually, each one a fruit from each plant, but it's really hard to ask this to them. So maybe in the following generations where we will have families derived from the selected plants in the first year, it can be done this. So we can add sensory, fast sensory analysis from farmers in these families. No, Andrea?
Yes, yes, I agree. As we said before with Mathilde, one taster cannot taste more than 5 tomatoes in one trial. So I think that when we have a few selected genotypes or lines being fixed, we can do something on this way. Yeah.
And if I can add something, yellow and orange tomatoes, you may lose all the volatiles which are derived from lycopene. And so you will have some specific, some other specific volatiles more derived from carotene in the orange ones. It will be interesting to look at. And do you plan to have one day a genotyping of your materials once they will be selected to see how they evolved?
No, this has not been planned in the task. Actually, the idea could have been of genotyping a few markers or a few genes for resistance, which could have been included into the population. But till now, we didn't plan any genotyping in this task. This particular one, not.
I don't know if is there any possibility in Work Package 4 to add some genotypes, perhaps at low density. We could discuss this if necessary.
Maybe what you should first look at the diversity, the initial diversity, because maybe it is very low, so more difficult to genotype at low density.
Yeah. Yeah, but at high density is more expensive, and I don't know, because in Work Package 4 there are a lot of materials to be genotyped, and I don't know exactly which is the situation.
I had just a remark for Joan. If I saw well, your questionnaire was not translated into French, so we could do it if you want, and try to disseminate it in France.
Mathilde, this was a mistake from my part, because as you know, my family, they are French. So my mom, she translated. So it's a right.
Okay, good. It's better. Yeah. But if you send it to me, well, if you send the link, I could try to send it at least in the University of Agronomy, because we have some regular talks, and also maybe to farmers interested in tomato.
Much really the dissemination of the survey. This is a hard work. I have made some tests last weeks, for example, I made a test with the students in my class. I had 30 students, and it was mandatory for them to do the survey, and I asked it to them to make a dissemination. So they say to me, "Oh, Joan, in 1 week we will achieve 900 answers," and the final result is that they had just 60 answers. So the 30 answers that it was mandatory for them plus just 30. So really to disseminate the survey, you should be persuasive. So send it via WhatsApp, because the survey, it can be answered via the phone, and also ask to students. Yeah, it's a continuous work and an effort, no, to move it. So thank you, Mathilde, and all of you. If you can help, it will be really helpful for us.
Joan, do we have a short presentation of the survey to use in dissemination to explain the aim of the activity?
I don't have produced it yet, but I will produce it. I think now it's time to start to produce some dissemination documents for posting in the web page, or as you are suggesting, maybe a PowerPoint, few slides just to explain the objective. Yeah, I will do it, Andrea.
Yes, we can talk with Workpackage 2 to arrange this.
May I, from Yulin? I like the comment from Andrea about the genotype of these cultivars or landraces with resistant gene. I would like to link to the work package 5, because we would like to monitor what kind of resistant genes are used in cultivars, commercial cultivars or landraces. So in this case, it would be very nice to genotype with a few marker genes for certain resistance genes. If you cannot do that, we can help from work package 5, but we do need DNA of leaf samples.
Okay, I will keep this suggestion for the next field.
Okay, yeah, so just let me know. We can also collect leaf sample there, or you can simply send us leaf sample, but DNA is better to send maybe than the fresh leaf sample or frozen leaf sample. But keep thinking, connecting this to work package 5 for resistance. I think it's a good characterization to show farmers which resistance is there in the cultivar.
Fine, sure. We will talk about it, and we will do it.
Okay.
And
yeah,
I think that we should also connect somehow with the work package 7 for this, for what Mathilde just said. And yeah, they are planning sensory tests, so we can interact and try to to suggest each other about this.
Yeah, thank you.
Any other questions? I think we can use the dissemination channels in work package 2 that includes not only other partners in other research projects under the genetic resource umbrella, but also the stakeholders groups that we have identified. We have already a large number of people that have contacts that we cannot, but we can also use other stakeholder groups from other genetic resources projects. So we can spread it. Because we don't want to have only academia and students involved in the survey, right? We want people and maybe not having higher education, because this doesn't represent the whole population.
Yes, yes. Okay. Regarding this, yeah, we want, we would like that participants are balanced between students, farmers, chefs, consumers. Like this, we can make a comparison between preferences from each group. So I think each group has a different channel, no? consumers, maybe social networks, farmers. I think you should just call them and ask to answer. I don't know, maybe also people from ThetaX who are really close to farmers. I think they will help us in this.
So I have a couple of comments. It's about the differences in earliness that you detected in between Spain and in Italy. Is this expected? And also the differences in the farmers' responses. I mean, with these two graphs that were complementary or like twisted a little bit. No, is this an explanation for that? So farmers are different and countries are different, because this is not about latitude. It's about when you started cultivating. I don't know.
So for the earliness, I would like to look at the meteo data of the season, of the growing season, but I couldn't do that because the regional system, all the regional system has been attacked by hacker a few weeks ago, some weeks ago, and they are still in trouble with some service and some websites. So I don't know if Joan got his meteo data from the fields or not, but you did. Okay. So as soon as I got mine, we can compare and we can answer this question about earliness, because I think that the temperature regimes is the first thing to look at. And about the farmer response, I don't have an explanation. I can only say that we would like to do this regression, and Joan is very happy to do them for many traits. So maybe we could have a better idea after that. And but don't forget that the farmers were not very much. So we don't have a very high statistical power on that. But we will keep you updated, and I think in the 18-month report we can tell the whole story for this year at least.
Okay. If any other comment on this for the work package 8? If not, we move to the next one. I don't think Annette is connected anyhow, but if she is there, just please say something. If not, we move to the next work package. We are just right on time, only a couple of minutes late. So the next work package is Mara with the global assessment. We just give 30 minutes because they haven't done much of our work at this first period of time, but it needs to also to transmit what they need to implement the work package in efficient manner. So please, Mara, if you can.
Okay. Good morning to everyone. I'm trying to share my laptop. It seems that there are problems with the computer. Okay.
Mara, if you like, I can launch your presentation from my computer.
Okay, thank you, thank you. There are problems for accessibility of Zoom on my computer.
Okay.
Yeah.
Everybody can see Mara's presentation?
Well,
yes.
Yeah, it's
yes,
on the
we
screen,
can see
Mara.
it.
It's on the screen.
Actually, I cannot see it. So what is happening?
Yes, I see.
Mara, if you go to the main screen of Zoom, you will see your presentation normally.
Oh my God.
Mara, would be good you open your own presentation and then we can see what is shared here.
Okay. It seems that the computer is blocked. Sorry, one moment, because I cannot go inside. There is this accessibility window that is here. I cannot move it. Okay, let's close this one. Let's see if we can see.
Another possibility, as I say, Yulin, is to open your presentation on your computer. Everybody here can see your presentation that I lost from my computer. And when you move to the next slide, just you say to me next, so I will put the next slide for everybody.
Okay, let's try this way.
Is it clear?
Yes, yes. Okay. So
now everybody see the first slide.
Okay, yes.
Presentation.
So I will talk about the VP9. This is the last work package of our project. Is on the global assessment of advanced material marketing strategy and life cycle. And I will lead this work package together with Semiorto Sementi. Please on. The participants to this work package, besides Junina and Semiorto, are mainly involved Enea, is mainly involved in Enea, and there are a lot of other participants with a few months, but it's very important that they will participate to this work package because as is on the assessment of our products, we require some interaction and some sharing of information. In fact, you know, this work package is focused on the assessment of project achievements and pre-breeding material. And this work package is organized in 3 main tasks. The first task is more an evaluation of project outcome impact on seed company, producer, and marker. The second one is more on from an economic point of view. So during the writing of the project, the leader of this work package, this task, proposed to develop a business plan and a definition or roadmap for commercialization. And now, looking to the project, he proposed to make a more broad economic evaluation of the novelty introduction in tomato seed market. And for the third task, so we will have an environmental evaluation through the life cycle analysis. Please, next. So this work package is just starting now. I will start with the task 9.1. And I am leading this work package, and there are some other partners involved, 6 India and Mercy. And in this work package, we'll assess the material and process developed in the project and we investigate it. So we collect information related to key outcomes, including classification, application, industry chain structure needed to implement it. And the next, what we are doing now is to looking at the most appropriate outcome descriptor testing questionnaire to make sure that they work for our purpose. And we are examining the list of materials and processes that are related for the project. So mainly we are looking to deliverables from WIP9, WIP3 to WIP7. Next. As for the search of the descriptor, the first step for us was to try to define the innovation. So have precise information about the goal of innovation. Okay, so for instance, an innovation could be to reduce the time to obtain a new variety, to benefit from an early result during the breeding program, or to provide information that can guide the breeding choice. And then we have to have some information on technical characteristics of this innovation, as well on techniques, material needed to implement it. And finally, we have to know who can benefit from it. As for descriptor, they are related to the evaluation of the impact in terms of improving the products and process respect to the current product process. We was thinking that could be good descriptor availability. So understand if this innovation is ready to use, the performance of the innovation as a degree of effectiveness respect to the current practice, the reliability, so the ability of innovation to give a result without failure. For instance, if we are looking to a prediction system, we have to know if this prediction is done in an accurately way. Next. And then, of course, we need some descriptor that are helping us to understand in which context this innovation is offered. So we have to know how we have to implement the program. So taking into account what we have to do before and after we apply this innovation. And there are then a lot of organizational aspects that should be taken in account, as equipment, infrastructure, personnel, and economic support needed. So all these information are very important to make an evaluation of our outcome. Next, please. So based on this consideration, we start to develop some templates and questionnaire templates. There are very simple templates. So we make a section where we ask for information, participant information. Then there will be request of description of innovation, which proposal is made, which are the technical advantage, information about significant factors that can contribute to implementation. Maybe we will add an additional section to ask if there need some additional component in the evaluation that was missing. And based on the input that coming out from the last WIP9 meeting that we had at the beginning of this month, we decided to make this analysis in 3 steps. In the first step, we will send a survey to all the principal investigator of our project to know about the innovation developmental status. After that, we will send a format to the company to know about company needs. And then we have to build up a model to evaluate the implementation and organizational aspect. So this will be the 3rd step. Next, please. So here there are some example of this questionnaire that we are making. Actually, a very simple questionnaire. So this is the one that we will send very soon to all the PI of the project. So be aware from that. You will receive this this survey. We are making in online form, so will be very easy to use. And what we will ask you? We will ask if you are developing innovation, which kind of innovation, of course, which is for, if is ready or will be ready soon, at least before 3 years, because it's the time that we have for finish the project. Who can benefit from this product? If it's difficult or easy of implement, and then we will ask some other information related to the reliability, if it's reliable or not, and the utility, it is available, is effective, have any risk. So these are the questions that we will ask you, and these will help us to understand which are the product and process that we have to select to judge. After that, we will send some survey to the company that participating to the project. And even for them, there are very simple questions. We based on the previous survey, we will know about the innovation available. So please change the available. So we will ask them to which innovation they are interested. If the innovation that are listed reflect their needs, if they are interested in other innovation that are not listed. Okay. Next, please. And then I told you, we start to give a look to the list of potential of products and process that we are developing in our project. Actually, looking to deliverables, we have a lot of potential products. So we were looking to the one that could be more interesting for practical breeding. So we start to select some, for instance, data visualization tool, treatment to reduce the generation time, and protocol for speeding breeding, prediction model, marker linked to resistance of other traits, and of course, lines that were improved for important traits, nutritional traits, quality traits, also other kind of traits. At the moment, we come out with a list of outcomes that we have from the end of this year and next year. So when we should make this our evaluation, and we get 5 of these products, a process that should be released. We are thinking to use them as a starting point for evaluating the implementation needs. Please, next. Next. So these are the ones that we are starting to think about, okay? But before making our final evaluation, we will develop in the next month the survey for PI. We think for the end of the year we will have this result, and based on this result we'll send a new survey for company, and then analyze all the data. We start to test the template for implementation needs, and for end of next year we wait to evaluate the first results. Okay, next slide, please. Okay, so these are the planned activity for month 12 to 24. Next slide. As for task 9.2, I told you there are some changing that are requested. So in the first version, next slide, we had to develop the task leader have to develop a preliminary business plan for most promising line, and to assess them, to develop some strategic guidelines for efficient and sustainable commercialization. Then the leader of this task, that is Semiorto, asked to change this the activity because in this way we can have more result for all companies that are participating to this work package, that are all the companies that are participating to the project. Okay, next slide. So what they propose to make an extensive study of the European tomato market, evaluating different outcomes that are obtained in the previous work package. And in this way, the broader study will be used to embrace the objective of different companies that are participating to the project. Next slide. So the proposed activity will be based on data collected from the previous work package. In particular, they will be focused on material with resistance to emerging disease or other biotic stress, material with enhanced nutritional and flower composition, and on tools that increase the speed and the precision of tomato breeding. As well, the predictive material that will be developed from traditional tomato varieties. Next slide. So what they propose to do? They propose to study the current state of European tomato market, to collect the data from the previous work package, and so to collect also market data and contractual terms of seed company, with respect of the broadest possible distribution and benefit. And then at the end, they will assess the impact of market of the outcome obtained in the project. Next slide. And as for task 9.3, the life cycle assessment, this task will be focused on the assessment of the environmental impact of products and process, comparing them with the business as usual. It will be focused on LSEA from breeding to seed production, on the full process, or will judge some environmental profile of our products. And so the focus will be on the product. The work package is led from Enea, but even in this case, there are several other participants. Next slide. So what is important for this task is to define, to well define the goal and the scope, because based on that, then they have to develop a life cycle inventory that should be very precise to make a very good impact assessment on the environment and to have the right interpretation of the data. So just as a sample, if we have a process, we can divide the process in different steps. So in each step, starting from the first one, they need information of the operation that we perform that could be impact the environment. And so here there is an example that is from manufacturing. So from the raw material, packaging, distribution, and so on. Of course, in our case, could be different, the step. Next slide. Next, okay. And to work well, they need to select the relevant products for perform this LCI. So it's important that we establish soon how many case studies to use. When they establish the case to study, it's very important that they establish a collaboration with people that are working on these products or process to know about the flow diagram of a process, because they have to collect this kind of basic data. For instance, if we are comparing a phenotypic selection against a genotypic selection, for phenotypic selection, we need all the data of input that we use during the selection, so fertilizer, pesticide, water use, energy consumption to produce seed, and so on. Instead, for a genotypic selection, maybe at some point, we have to use plastical disposal in the lab, reagent to perform PCR, make DNA extraction, and then maybe we have some consumption of energy for computational analysis of data gotten. So we have to be very precise in each process, which will be the need, the input that we use. And as I told you, we will ask for a deviation in our work package. We asked to modify task 9.2 to guarantee a wider impact of the result obtained from Arneson. Next. Next. Of course, making this modification, we have also to modify the deliverables for 9.2, that will be not anymore roadmap to commercialization as SWOT analysis of most promising project output, but guidelines for novelty introduction in European tomato market. Next. So we have not yet dissemination and training activity. Thank you very much for your attention, and if you have any question, I will wait for them.
Yes, Mara, can I ask a question? I didn't understand if at the 9.3, the life cycle assessment is at the level of tomato variety, which has already been developed, or will you do a life cycle assessment of pre-breeding activities?
They can do both. They can do both. Actually, they have to define. We have to find a few cases. If you have some case that could be interesting, please, you can contact Sara. Sara is here. Sara Corrado is the person that will be involved in that.
Yes, I'm here.
Okay. So Matilde is asking if we can evaluate a variety that is already developed. Matilde.
Yeah, no, my question was more general. If the life cycle assessment will be on variety or on which kind of product finally? Is it the pre-breeding activity?
No.
Or the once the variety has been developed, then all the cycle of the production?
Well, please, Sara.
Yes. Well, I think this has to be decided for sure. For the SCA, we would need data on the production of the variety and on the performance of the variety. So it depends if this data could be available also before the development of the variety, kind of let's say a projection. I mean, not real data, but some assumption can be done. Then we can decide when to perform the SCA.
Okay, thank you.
So we have to decide. She is not from, let's say, our environment. So she's working on more on this field of environmental evaluation. So we have to tell her few other cases that would be interesting to evaluate it from environmental impact.
There was in the Traditon project, there was also some LCA which was performed by Enea. So maybe they could also use those data to continue and to complete those studies. Yeah.
I know it's my colleague who was involved in the Traditon project, so I can ask him data for that, or yes, or ask how the study was structured and how they manage the data collection. Because as I said, as Mara said, I mean, I do LCA, but I'm not used to apply LCA to this field. So for me, it's a bit difficult to understand what is more meaningful, to which product is more meaningful to apply LCA.
I think from what I remember in Traditon, the LCA analysis that was performed was with the help of Alcalá, for instance, where they produce, were given the information how they grow the tomato, traditional tomato varieties, and this was compared to greenhouse intensive production in Almeria. So basically was this life cycle comparison between varieties, traditional varieties cultivated in, let's say, in traditional ways, they do it in some places, and also in greenhouses, intensive production. Things like that. And I think it's nice that you can have access to this information, but in this case, I think the challenge here is to see the breeding. What is the impact of life cycle in the life cycle assessment of introducing a disease-resistant gene on a variety that has not such a thing, or increasing the tolerance to this or that stress. So it has an impact on the way you grow the plant or the amount of crop that you can harvest, things like that. It can be also assessed the process as we discussed, of course. Yes. Okay.
Yes, but it will be very difficult and different from the life cycle assessment, as you may not analyze it in relation to the yield of the product. Yes. When you're in progress disease resistance or any other thing. I think it may be discussed, but it may be very, very complex also to do LCA on pre-breeding strategy.
Yeah, it has to be a projection on the future. Let's say, yes. If you have I don't know, an increase, I mean, a QTL on increased flowering of fruit set, and that this circumstances of an increase of 2 degrees in temperature. So, and this could be projected on what it can be the effect. All the rest of parameters and inputs and outputs this being the same, except for the change. But of course, this needs to be discussed.
Oh, I think it is pretty straightforward. I mean, we have to decide what traits to focus on, whether it's disease resistance or abiotic stress resistance, and not wait for the newly bred material, but just, take some reference lines that companies have already probably available for this business and grow them side by side according to the same protocol and measure output and all the other parameters. So I don't think it's very difficult. Of course, Sarah has to discuss with Christian, who was in the Traditon project, how to collect the parameters, etc., etc. But I think the object is straightforward. As long as we decide what traits to focus on. So how many traits do you think you can measure, Sarah?
Well, actually, I don't know, because I think this is something that goes, you mean in terms of study, sorry?
Yeah, the size of the calculations you can make. How many field experiments, how many variables?
Okay, because I will assess the impact of certain products. So I will wait inputs from you on the types of products and number of products. Yes, I think, I think the number of traits doesn't really matter for the LCA and for the work that I have to do, because I will need information on the, for example, energy consumption, and then on all the materials that are needed to cultivate the tomatoes, so on fertilizers, pesticides, water, field work, so these kind of things. So I see, at least if I understood correctly, what you mean, that the number of traits is not really related with the LCA. Maybe what we should discuss is the number of LCA that we want to perform. So how many products and how many processes we want to perform. But also for this, I think we need to discuss to see what is relevant and significant for the project.
I think we follow the following course of action. Christian had prepared a questionnaire in which the growers were giving all the information that was needed for him to perform the LCA. The first thing to do is to get this questionnaire and send it around to the partners who are performing field experiments in order to see how many of them are making these inventories and are able to provide this information. And then perhaps we will see that the amount of partners that have a very accurate inventorying of the inputs and the outputs are not so many. Many of the partners probably just spray insecticide without measuring it, or give fertilizer whenever they remember to do it. Some others probably have a much more accurate way of accounting for these things. So the second step is to choose these partners that have this accounting method that can provide reliable data. And then the third step is we see what traits are these partners working on. And then at this point, the choice of the trait is sort of obligatory, because we don't have so many partners that can. It's much easier to go to a partner that already knows how much fertilizer puts in, at what distance, how much water, how much energy in case of greenhouse growth. Okay. So the first thing to do is, I suggest, you get the questionnaire from him and send it out to the partners that are doing field trials and see who is able to provide the data. That's what I would do first. Okay.
Yeah, I agree.
You don't really need, in my opinion, maybe others have different opinion, you don't really need to wait for newly bred lines, because there are already lines that have traits in them. I mean, there are lines that are resistant to a certain virus, there are lines that are resistant to fungi, there are lines that are resistant to heat. So there are already things there that can be assessed from day one. Not from day one, but from the next field trials.
Yes, this is true for the lines, but then we have to evaluate also the process. So for this kind of evaluation, maybe we have to choose the.
So it is the process or the lines? I thought the process, the growth protocol was Traditom, right? Then
What?
we evaluate the traits.
Giovanni, the question is that there are processes here in this project that maybe that's the question that also Mathilde was asking, whether we are talking about pre-breeding or processes that speed up breeding, for instance, that need to be evaluated not for the end product, but for the process and the way they affect the breeding process. So it's a different kind of thing, and it was part of the work program.
Extremely complicated.
No, I know the easiest way is to revisit it, something that could have been done even before the project, like you are suggesting. We take varieties that are already in the market, that there is already experience in growing them, but this is different.
No,
And it was done already in Traditom.
It was done. And the partners that were participating in Traditom, providing this data, are also part of Harvestom. Is Catalunya, and Joan provided data, was Andrea providing data, and was Alcalá providing data. And out of the three, only Alcalá was the actual farmers that they produce tomato and grow tomatoes to sell tomatoes, while the others are academic partners growing for breeding or for scientific research. It's different settings. So I guess the life cycle assessment for a product that is in the field is a finished product. It makes sense to do it in a commercial setup, in a commercial facility. Then you can collect all the data that is needed for the life cycle assessment, and you have this. But in the context of this proposal, that is a pre-breeding proposal, it may make sense as an exercise to do a pseudo life cycle assessment, either on the cost of producing something, of breeding for something, or developing a process that is useful for getting a process into the breeding company. It's a different thing.
I'm very doubtful that you can do that. Let me give you a practical example. You could compare phenotype-assisted selection versus marker-assisted selection, okay?
Yes.
But the companies that do marker-assisted selection, probably they are not willing to run in parallel a phenotype-assisted one and collect the data, because marker-assisted works so much better. So you will not find a partner that is able to give you the comparison between the two methods. They are using either one or the other.
Yeah, but you can have some information about the means. They know how they perform before use marker assisted selection.
They performed in a different environment, was maybe 5 years ago, they used different personnel. So it's not very comparable, really. We all know that if you want a good control for an experiment, you have to do it in the same conditions, right?
Yeah. Okay, I think this deserves more in detailed discussion that maybe we can do now. It's already a quarter after the time, and I think it deserves maybe discussion whether we stick to the program as planned. We would make more changes. I mean, I think this. The work packet is already introducing some changes to the initial program, and I don't know if we can do more changes, additional changes.
Yeah. What I would, a possible way to do it is, okay, you have a, let's say you have a disease resistance trait, and you have a abiotic stress resistant trait. So what you could do maybe with the life assessment analysis, life cycle assessment, is understand how much improvement, economic, environmental improvement, do different types of traits insert.
Yes, that's what I think was what Mara was suggesting, and this was part of the change that
you could, I don't know, you could, if you have them, you could take isogenic lines with heat resistance, virus resistance, etc., grow them side by side and see and see what the output is, what the inputs you have to put in, etc., and compare.
I mean, I don't think we can do this in a commercial setting. We cannot grow transgenic plants versus some in comparison to anything. We just do that. We have to do the academic exercise, like in silico, using the actual data that the farmers' inputs and output they get, but without actually growing the variety. Okay, that confers the added value, the increase in 10% production, for instance, or the 10% increase in fruit set, things like that. It's the only thing that we can do. But anyways, this.
Okay, let's discuss.
Yeah, we can be discussing this over dinner tonight. Yeah, okay. Let me just go to the next presentation. Is myself. It's about the ethics. The Work Package 10. Okay. Simon, are you there or go for lunch? It's time for lunch in France, right? Are you there, Simon?
It's coming. It's coming.
Coming. Lunch?
Lunch will be later.
Okay, I will go fast and smooth through this.
Okay, you can see my screen?
Yes.
Okay.
Okay, next, this is about the ethics. It's Work Package 10, as you know, was not in the original work program that we sent for evaluation and, to the European Union, but at that time I filled in a self-assessment on ethics. And this means I was trying to say, okay, we don't need a specific ethics work package, but after the review of the proposal and the questionnaire that I filled at the moment of submitting the proposal, there was an ethics review, and they said, no, you have to introduce an ethics work package. Can you come back one slide? One forward, okay. So we need to introduce this, and we did it. And basically, the requirements were to fulfill a number of deliverables that we have to do. And this means that we all need to comply with what asked. And there was not personal month to be imputed in these work packages. Okay, like fantasy. You have to do work, but you cannot introduce personal month. You have to do it without much of physical or mental exercise, what is impossible, as you know. Okay, so there was the objectives of this work package on ethics is to comply with the regulation on ethics. That the European Commission say, okay, well, this is a legal obligation. It's not that you have to do something else that you are not to do, because this is something that is an obligation, but you need to produce these deliverables on time and this work package. So there was a series of deliverables in what they call H, meaning health. It was another deliverable in GDPR, which is General Data Protection Regulation. This is what the GDPR stands for, because they identified that we were using humans as research objects. And I thought, well, how are we using humans? When you have a taste panel, we are using humans as research objects. And we are not asking these humans to eat, I don't know, alien food. No, it was tomatoes, but still, we need to comply with this regulation because there were opinions. And when you ask people for opinions, there's privacy. Now everybody's privacy is very important, although you can send WhatsApps and emails that are replicated all over the world, but still you have the right for privacy. Then it was the NEC, and I say, okay, we have countries that do not belong, are not European countries. In this case, we have Israel, that is almost European for many participators in European country in the EU projects, but they are not European. And we have Taiwan, although they don't receive directly money from the EU, they are funded from other sources, but still we need to comply with this regulation in relation with the non-European countries participating in the project. And then we have environment, health, and safety. This is EPQ, and and I say, okay, health, environment, how we are growing tomatoes, but then we also, some of us are doing edited plants, some of us are doing, are importing seeds, sending seeds outside. We're also using wild relatives, and then we have all these issues about using germplasm and then sending seeds around with possibility for introducing diseases in other places and things like that. So this was basically the idea. Next one. So the health requirement. Okay, the health requirement is that we are recruiting or people for participating in some of the work packages. And this is specifically in work package 7 and 8. Work package 7, you remember, is quality, and work package 8 is the participatory breeding with consumer chefs, taste panel, and so on. And work package 7 is about health and quality traits, including sensorial analysis of the yes. So we need to have informed consents. We need to produce templates for these consents. And this has to be, this is the requirement for humans, is that we need to submit a deliverable, and all the informed consents and all that forms and so on have to be written, of course, in the languages that we are going to have the panel. So we have to produce this in French, Italian, Catalan, and Spanish, and keep them on file. But we need to produce the deliverable that we have done our job. And then we need to have the ethics committee approval before we start anything, any activity. Nothing. It doesn't work after, and we know this by experience. That is very difficult to get an ethics committee okay several years after you have done the experiment. Maybe even 1 week after, I don't know. But several years is very difficult. Okay. Next one. And this was basically for work package 7 and 8. So what happened is that we ask each partner to contact each partner involved in any of those work packages to contact their ethics department, and then to collect the information about the consent templates and so on. This was done on time and still ongoing because not everybody has already get to the point of having to deal with humans as subjects of research. Next slide. Then it's about how you keep the information, and this is the deliverable, the 10.2, that has to do with how we process this data. And this again affects basically work package 7 and 8. We need to have an appointed data protection officer. Normally, academia groups have this in their institutions. But then we need in the deliverable, we need to explain how we intend to process the information, how we intend to keep this information as simple as possible, how we are going to keep the information related to the human not linked in a file with the results of the human, let's say, participating in the project. Okay. There are a couple of points that didn't apply to us because there was not transfer of personal information from the EU to non-EU countries, because neither Israel nor Taiwan was participating in these questionnaires about consumers and so on like that. Okay, so next one. So deliverable 10.3 is about non-participation of non-European countries, but also about the use of materials from non-European countries. And because we use wild relatives, we have to comply with this. Okay? We need to know also about the import-export authorization are required by each national legislation. And we need to keep this on our file. This has also been very active. We also need, and this applies for work package 4, where we have lots of population that derive from other countries, but we need to see whether we are in the situation to report this or not. And then we in work package 5 and 6, we are basically working with materials that can introduce wild relative germplasm into domesticated. So next one. There's about environmental risk. We have some activities using viral vectors for speeding up flowering. We have some activities for increasing recombination rate using CRISPR. We are also using some viruses that are quarantine viruses. So we need to have all the appropriate and necessary health and safety procedure in place, and also to comply with the legislation. And all this has to be submitted as a deliverable, and we need to have all the forms and documents kept on file and used when necessary for sending material. Next one. So what we have done then is that with all the partners that according to the work plan need to exchange plant materials should be aware of the EU regulation, and all these regulations have been compiled in the deliverable. And then all the necessary certificates for sending and exchanging material have to be obtained in this kind and reported. And I just ask you, because I know you have already done that to send to CIMO, and CIMO will enable a place at the repository to collect copies of all the documents in case we are asked to provide them by the Commission for the Ethical Committee. So CIMO will develop a specific place in the internal repository of information in Hammerstone, and in the management section or in the general section. So we can, not only we have a repository of papers published in the consortia or the reports produced, we have an additional one about the formalities and we have to do, like the phytosanitary documents, the import-export docs, okay? So please do comply with this. Just send a copy to CIMO of everything you use to exchange materials.
Okay,
and also
just an addition, Tony.
Yes.
Normally with the login and password that I sent to the PIs,
Yeah.
they can access very easily to the Dropbox on the platform. They just drag and drop the file. But if they want to send it to me, it's no problem. I can do it. Okay.
No,
I will send an email
want-
just to explain how to do. Okay.
Yeah, but it's be good if you inside the Dropbox you put some folders.
Yes,
so that
yes,
it's
yes.
not like a
It will be
box,
connected to
like
the
box
folder.
with
Yeah,
folder.
yeah.
So you produce the folder and the people
Yes.
will just fill it.
Exactly.
Okay, sounds good. Next one, please. Again, for the 10.4, all the partners that need to use infected material or inoculous pathogen for the screening should be aware of the EU regulation or the national regulation and report them. So work package 4 and 5 with all the information on what measures they will take to mitigate the risk in case the pathogen escapes their control. So, and this was already reported and it was introduced in the deliverable. Also, Felipe, our legal partner specialized in the legal issues from the UPB, updated and produced all the legislation that we have to comply with. Okay, so and this was introduced in the deliverable. Next one. Again, the copies of the permits for the relevant facilities to use the pathogens or to use GMOs or edited plant. We need to have the copies. If it's in your own institution, it's fine, but it's even better if we have a copy in the repository that CIMO will be putting together and offering in the Dropbox. Okay, and this affects work package 4 for the editing and work package 5 for the evaluation of the viruses and new strains of the Cladosporium fulgum. Okay, next. So please send this permit so that we have a copy. Okay. About the environmental issues, there was also a requirement that we demonstrate that the appropriate health and safety measures conforming the local national guidelines, followed by the staff, and it was also produced and submitted as a deliverable. Of course, all these submitted as a deliverable can change and be updated. So if there is additional experiments that require additional permits, and exchange of seeds are produced in the future, you have additional, we need to keep a copy of this. If necessary, we can update the deliverable. Next one. The 4 deliverables were on time, month 3. I put them together with the feedback that I got from you. The deliverable 1, the requirements on human, the deliverable 2, requirement on how to process the personal data, the requirement 3, how to deal with the interaction with non-European countries, and then the deliverable 10.4 for the environmental risks, and it was done. Okay, good. Next one. I don't want to spend more time with this because this goes back in time when we have to modify the grant agreement with the work package 10 added, and it was done. But this was earlier, and so maybe this slide that is out of place here. Next one. This was how the process was conducted, and it was activities in these 2 months. The partners were contacted, information was retrieved from them, all them agreeing to comply. Then the partners in the specific work package provided all the information, and then all the consent permits, FITO, and so on are stored and available. That now I'm asking you to be stored all this doc in the repository in the arenston.eu. Next one. Next one. Okay, this is details that I don't need to go back to you because the deliverable was already observed, is already produced. It's about definitions, but there is no time now, I guess, and it's more important that we go into maybe some discussions for other issues. So please go through the slides in case there's something I want to say or highlight. Simon, can you move forward the slides? Because this was just to remind you about anonymization of the data, about how to process the data. But this was already indicated in the deliverables, and all the people who are already or are gonna do these questionnaires for the consumers or farmers, they already know that they have to comply with this because they are already putting together the questionnaires and the consents, so they are aware. So there is no further need to go through this. Move forward, please. About data minimization, this is important. We always like to know more, but this is like Miami time to approach. We need to know as much as is needed for the purposes of our proposal of our project, no more, no less. Okay. Next. This is just move forward faster because this is just. I don't think this is necessary about. Yeah, this is about a little bit about the reason why we are asking to comply with the regulations for use or not genetic resources. You don't have to forget. You need to remember that we are in a proposal what is about genetic resources and the use of genetic resources. And there was already a number of meetings at the international level about that. If we use natural resources, we need to share the benefit of using this. And there's a lot of regulation in relation with this. But if you go to the next slide, basically we are asked is in this project we're using genetic resources that were acquired after October, 12th of October. Is this, by the way, when Christopher Columbus discovered it? No? 2014. And apparently most of the materials were in our facilities before that, so we are not on the scope, in the scope. Okay, so you can do without doing this additional processing of the requesting this going ahead with this. Okay, so in the case that there was the case, then we have to fill up and upload this information here. Okay, but I don't think we are in the scope of this regulation for the project, as far as I know. Next one. Next, Simon. Next. This is about information. The presentation will be, and I remind you to send Simon, those who may have not sent their final version, send Simon the final version of the presentation, so it will be stored also in the intranet of the project. And this will be also, in case you want to know more about the ethical issues and so on, there are links that connect directly there. Next one. Okay, so just as the summary of the deliverables, all the research participants' informed consent and corresponding templates to be used were defined to ensure the compliance with the ethics requirement, and all this was produced in the deliverables. The approvals of the ethics committee indicated, the informed consent indicated, and templates produced basically by UPV, UNINA, and UNITUS, that are the ones normally dealing with this. We may need for the stage panel, if we do this in outside of this institution, we may need additional ethics in the other partners that may need an ethical a panel assessment of consumer panels. Next one, this is the 10.1, the 10.2, that is the measures taken to protect personal data of subjects involved in the research and comply with this requirement. In all these, the main three institutions that participate in tasks that require this, the data processing officer was appointed, the explanation on how to process information was indicated, the description of the technical and organization measures to safeguard the rights and freedoms of the subject were described, and the security measures were described, and the anonymization and pseudo-anonymization techniques were described in the deliverable, thanks to the participation and help of these three institutions partners. Next one, about the non-European countries' participation. We basically detail the materials to be imported and exported from the EU, with a list of copies of import authorization are required by national legislation being kept on file. This basically applied for many of the materials that Peter Hanson is sending around, and I think he has followed all the regulations. And in today's presentation, there was the list of the different requirements for each of the shipment of seeds that he has or is planning to do. Then there are a number of annexes to the deliverable, that is like the model of a standard transfer agreement, the regulation that apply to each of the participating institutions, the tables of genetic resources that will be used in Harnestone, and a flow chart of how we use it, and we will change the materials in case it falls or not in the scope of the regulation that, as I said, we believe not. Next one. Next. So this is a snapshot. Next one, or previous one. Previous one. Yeah. This is a snapshot of the diagram that we find that have to follow to see whether we are in scope or not. These are most of the materials that we are using for some of the pre-breeding CRAM come from previous project, Traditon, Tom Jumpress, of GTP Soul to Power, and this was previously funded by the EU with the contact names here. So we are not using new material imported, but material that were already even part of the previous EU project. Next one. So we are out of scope. This is the final thing, the final one. So it's about the environmental issues that we provide information that we are not going to cause any harm in the environment, but we are going to take all the measures of containment to mitigate and also to mitigate the risk, especially when we are dealing with quarantine viruses and things like that. That we will comply with phytosanitary regulations, and we have all the papers and docs and permits obtained and safe. Okay. This is just details on that, and some snapshots of tables where we describe the use of pathogens and the screening for resistance in the different locations and different partners, and what is the purpose of the experiments. So move on, and then
the list of authorizations that we have for use in the different laboratories and to do the screenings or to use plants edited or GMOs. But again, we need a copy of that. So this is the final slide, is that all deliverables have been produced and uploaded, that the ethics committee approvals are obtained, are kept on file. I just need you to keep them on file, but in the European, the Harrington website. The same for the informed consent. Whenever they are produced and obtained, we need to keep them on file. All this information is available, including an update of the legislation that was produced by Felipe, Work Package 2. And then all these other phytosanitary certificates and certification also. So this, I remind you to put them in the enable repository. Okay, and I think that's it. Okay, so we are almost back on time, but I didn't get any feedback from you to have specific workshops, but we need to do this for the annual, for the 18 months meeting. This, and so maybe we can discuss some issues that are pending. Okay. I want to remind Simo again, not only the repository, the folder for these docs, please send the Excel file or a Google's doc so that people that join the project after the initial kickoff may add their name and email so that we have a complete list. So when you send the general email to all the partners, we send a general email to all the partners, so are all included in the list. Okay.
Okay.
It's important that we all work with a management platform that is a very useful tool. And I ask you to start putting content in the management tool, specifically for the task, for the PI leaders, for the IP, for the leaders of each group in each task, also for the task leader to do their part, and for the work package leader, so that this will facilitate when we have to write the progress report after month 18. Okay, so please start filling with content. So this time we didn't have to produce an annual report. It will do the job, the recording, the video that I can see that Simo is doing. So we have like a live talk in which we can go and visit in case we have some doubts. But because we have to produce a report, it's better we start already writing down the things that we just presented today and yesterday.
Okay. Also, I will put the video that we made when we had the training, so on how to use the platform, how to report on the platform.
That will be useful.
So it will record you how to use it. I will send you this just after the meeting, the link for all the presentations, so you can have a very small training how to go to the platform and how to download the files if you like. You will find inside also all the presentations, previous presentations during the kickoff meeting and other documents. Also the ethics document that just Tony said about. Okay, and I will give you the link to the Dropbox that is linked to the folder when we have to put the ethic documents coming from each partner.
Okay. I think it's important that we work as a collaborative project. I mean, we facilitate the work of the work package leaders and the coordinator, and also that we respond as quick as possible to the requests that sometimes we do as leaders of work packages or coordinators. For instance, in the case of work package A, to participate in the dissemination of the surveys because they need our help. So we all need to help them. Also for the other, for instance, Mara was asking for help from us to indicate what possible materials or processes could be suitable for this evaluation of LCA or whatever. So as soon and helping to designing the surveys. So, but not only, for instance, work package aid, all these surveys that they want to distribute in different countries. They need help in all the different countries. So I know this is not, this goes out of our lab, but we have, for sure, we have list of emails we can use also to distribute these surveys. Okay, so try to help others in their activities whenever it's possible. Also for Work Package 8, the dissemination is, you remember that Maria Jose indicated that they have like a plan, dissemination plan for Twitter and so on, so that each of the participants in the Work Package 8 have the commitment maybe once a month or every 6 weeks to produce a short news that it is sent through one of the media. So that's keep us people aware that we are working with the project. Okay, that's good. So this is our general issues, but I would like now to go back to when we have, and I think in general the presentation have been perfect. I mean, I think we have been, I mean, each work package has presented nicely the progress made, the possible problems and so on. And I think we adjust exactly almost to the time that we have. So we are on schedule by the minute almost, which is very good. But I think for the review meeting, and I will ask you, Simon, now to put the pre-review work PowerPoint presentation that I sent you yesterday. So for the review meeting that is due after month 18, it's better that rather than each partner presenting the little piece of contribution to each of the work packages, that this is led and presented by the work package leader, because otherwise it will be difficult. We aim to have a face-to-face presentation or interaction with the reviewers and with the officer after month 18. Okay, so it won't be, that's the plan, unless things change dramatically to the worst in the next following month. The expectations are high that we can have a face-to-face meeting with the reviewers and the officer, hopefully in Valencia. That's my proposal. Can you put the presentation of the pre-meeting and review meeting?
Okay.
PPT. I don't want to forget, after this, I would like maybe to get back to with Giovanni about the genotyping, because I think this is good that we take the opportunity that we are all now together, because you sent a revisiting of the table, Giovanni, that maybe it's worth that we discuss together about how many things with different platforms. And you made a proposal by email last night or this morning, I don't remember. Simon, can you put the
yes, it's coming.
We are done with that and we move forward. Okay. So next things to do, apart from continuing our activities, scientific activities, is that we need, after month 18, the interim report, to produce the interim report. Then we have to arrange a review meeting with the officer and the reviewers. That will be probably 2 people that the officer will present to us, and we have to agree whether we have or not conflict of interest. If we are happy with them, then they will review our project. Of course, the review meeting has to be done after the report is presented. And I'm proposing you to have a pre-review meeting the day before of the review meeting. Okay, so that we get together without the reviewers the day before to have like a training session for the review. So and then we can also revisit what we have done in the last 6 months after today. Okay, so move 1 slide more forward, please. Next slide. Simone. Okay, remind you that in Crediton, like in Harveston, like any other EU funded project, we have a continuous reporting module. This means that during the first year, we have been uploading deliverables and updating the data, the portal with new things at any time that's needed. But after month 18, you have to report. And the report has to be produced within the next 60 days. So we have 2 months from month 18 to month 20, okay? This means that April, May next year. We can start, of course, to prepare the scientific report month 17 to have it finished end of month 18. That would be the best. Why I'm saying so? Because this is the sooner we report, the sooner the report will be approved, and the faster we get the next payment. Okay, next slide, please. So this is a possibility that in March, April, which is the month after month 18, we might, because we start to collect all the information before, we can have the team leader report in Friday 8, and then we have the work package leader report in day 14. So we have 1 additional week to process each of them. So by middle of the April, we are almost done with the scientific report. Okay, and this is 15 days after the 18 months. Then the financial report, sometimes it takes longer. No, go back, please. The financial report, we are 22 partners with different administrations, each with their own timings and things. Also, we have to keep in mind that Friday 15 and Monday 18, this is Good Friday and this is Easter Monday. So these are complicated times, but this is the time it is. I mean, so to avoid this, and we can spread a little bit more. Next slide. So rather than concentrate it on all the work here, we can spread to the next month. Okay, so at least the financial. So and we can move the dates. So I just want to get your feedback. We want to have to produce the final scientific technical report later or earlier. The earlier we have it, the best, because we get the review meeting earlier and everything goes faster. But in any ways, whatever we manage to do, my proposal for the review meeting, and we have to let the reviewers enough time to go through the report. And the report is quite a number of pages. Normally it's more than 60, 100 pages, even if we put them very succinct and straightforward. So next slide is my proposal, is that we finish whatever we have to do 1 month or 2 months after the 18 months. That means March, April, May. But in June, we have to make everything possible to have, if possible, the review meeting with the reviewers on the 7th of June. And the idea of proposing this is that if the officer and the reviewers are fine with this, then we can meet on Monday or even Sunday and Monday for the pre-review meeting. The scientist or the IP or your package leader or whoever we want to do, we can be all or only the work package leader. For the review meeting, it's better if only the PIs from each institution and the work package are present. It's not something that requires the participation of everybody, but at least the PIs and the work package leader. But in the pre-meeting can be as many as we like, all the participants. And it's good because in this way we can connect with the Eucarpia meeting that will be organized here in Valencia, the 31st, the 1st, 2nd, and 3rd of June. So if you like this idea, then we can. I already contacted the Scientific Advisory Board and they are happy to come this week, basically, starting the 31st and finishing the 7th. We have already contacted Lucas Miller. I contacted, who else? David Francis, Peter Hanson, Peter Hanson, no, sorry, Peter Hanson is part of our partnership. Sam Hatton and Roger Chetteler. And all of them are happy if the situation allows them to come and visit and interact. So they can be invited in part by Eucarpia, because they will be talking in the Eucarpia meeting, and part will be funded by Harnestone to come and stay with us for the review meeting. So this is my proposal to you, that we work on the elaboration of the report early at the end of the reporting period. Of course, the financial reports cannot be produced before the financial period is finished, because the administration doesn't like to do this before it's done. So, and then we have to cover until the last expenses of the previous period. But I think this is a good proposal, but I want to open this for you to discuss, okay? What do you think about merging the Eucarpia meeting with the pre-review meeting and then the review meeting? Anything pro or against it? And of course, coming to Valencia, those of you who, of course, the PIs and the work package leader,
Me is okay.
they must come, but the others are more than welcome, because this will be the first face-to-face meeting. And this is something that I think this what makes a project. I mean, you start to feel that you are part of something when you meet together. I mean, this is very unusual, at least for me. Okay, so if you feel like this is a good proposal, I will contact the officer to suggest that this is the plan. So, because she will need to find reviewers, send us a proposal for reviewers. We will distribute the names, agree with them or not, depending on whether they are our best friends or our worst enemies, say yes or not for the one of the case or the other, and then go with this plan, okay? But in order to feel, I mean, I am very much in favor of speeding up the review process as much as possible, because the longer it takes, the longer it will take, because there could be some reviewers that they want more information on changes, and this will delay things. And if things, so can you go next slide, please, Simon? Because there are some notes here. So, okay, this is the 4 years time. It's like 12 hours divided in four years. But the first 12 months is the 1 year. Then in month 18 is 1.5 years. We have to produce interim report within the 60 days after this. And then we have 90 days more to submission and approval. But don't forget that we have already got about 50% of the budget. So we can live with this money even for half of the period, because we receive half of the money. So we are, even if we manage to get the project review accepted by the European Commission and get the payment, we are still in a safe zone. Yeah. Okay. Giovanni is pointing that he doesn't like working on Sunday. I understand that, because he's from a Catholic country and he's living next to the big boss of the Catholic world. And I'm against
we all are from more or less from.
So we can do that like that. Okay, we can even move things a little bit to Tuesday, right? Tuesday, Wednesday, if you like. But so we can just take the Sunday off, or the Saturday and Sunday off, go around in Valencia between Eucarpia and the pre-meeting, and then we have the meeting with the officer Tuesday or Wednesday. We decided, okay? Everybody wants not to go to hell and respect weekends and holidays to honor our Creator. Okay, we do like this. Next one, next slide. No, there is no next slide. So I will start then preparing, informing the officer that our plans is connect this Eucarpia. She's also aware of the Eucarpia because it will be co-organized by us. And I take this opportunity also to thank Giovanni and Jaume and others, because we're going to have a European project meeting, workshop, right, Jaime?
A workshop, but when?
Oh, the workshop is part of the Eucarpia. No?
Yeah, of course,
the coordinators of the main funded projects on tomato, because there's a Eucarpia tomato to participate. So, and I think it would be good if we can use this event as a promotion of a dissemination activity of our respective projects. No, like it doesn't do any harm if we use the same event for several issues. I mean, to make people happy. Okay, officers and scientists and companies, everybody's happy. We give them credit for what we're doing. So if you are happy that we have the
just check that I have no conflict for those days, so I can come to the Eucarpia.
Okay. No, but you confirmed already, no? The participation.
I wrote to you that I had to check.
Oh.
Now I confirm that it's fine.
Okay. So we have confirmation for everybody that was invited. That's good. And then it will be all the tomato coordinators basically recently funded or already finished their project anyhow. So I don't have much more to say, except that do you want Giovanni to revisit the table that now we are all together, or you want to discuss this in an email basis?
If you wish, if there is appetite for it, I'm willing to do it. Maybe people want to think a little bit about it, but I think some things are, at least for the requests that we got, we can go through them again and try to, I think that's a good, at least try to move them up to resequencing or move them down to resequencing 10x or 1x. So yeah,
but maybe
we have 15 minutes, we could do that.
Because an important issue is how much of the 80,000 plus that you are now cutting the budget. I mean, you're going from 100 to 80, and you have budget for 50,000, right? So we still need to find how to cover this.
What I got is that also some other partners that are on this task have some budget for this. Is this correct?
Yes.
It is correct.
Yes, so, Tony, we had allocated 50,000 for genotyping.
Oh, that's good.
So
okay.
So as you see, I'm saving money, but in perspective, that will be used because
yes,
I mean this is just the second. Next year will be the second year.
Yeah,
and we will need more during the third and fourth year. So I'm sure we will find a use for all the money.
No, we have just to look to find the best way to use wisely the money that we have. I mean,
exactly,
with responsibility and doing the best and the most effective way.
I can open the table and go with you. Please. Do you agree?
Yes, but I want to set the 1:30 and we close and wrap up. Okay.
Yeah.
Is that okay with all of you? 1:30
plus 15 minutes.
Okay.
So, and I send it to also all the work package leaders, not only the work package for partners, so that everybody can see if they want to add something. So let me share my screen. Can I do that, Simon?
Yes, yes.
Okay. You see it?
Yes.
Good. So, increase a little bit. So basically, it looks like that we are going to simplify this whole thing. And my proposal at this point is provided we get a positive result from the test run we're going to do, and we have three basic approaches. One is 10x resequencing. Why 10x? Because 10x is sufficient, is already. So let me tell you, most of the pangenomes that are out there for tomato are more like 10x than 20x, like the one that was in the Gao et al. Paper is mostly 10x. The second thing is 10x is sufficient to construct a hap map. So if you have, let's say, a magic population with 8 parentals, and you want to genotype the whole progeny, to do this by 1x resequencing plus imputation, you need 10x resequencing of the parentals. Okay, that gives you a good coverage for getting a good hap map to do the imputation for the 1x resequencing. And the third reason is that 10x is sufficient for doing K-mer-based GWAS, which we might want to test sooner or later in our project. So my proposal is to abolish the 20x and to go for 10x. And I think we can get as low as 90 euros per sample. I'm still working on it, but I think we can go as low as that. Now, the advantage of 1x plus imputation is basically you do 1x. They do the library, they resequence 1x, and they impute the missing SNPs based on a database that they have in-house, which is already based on the public tomato resequencing data. We will add the G2P-Sol resequencing data to those, so this will be quite a respectable one. This will work very, very well for GWAS. For segregating populations like MAGIC or whatever, I mean, recombinant inbred or BILS, you will need to resequence the parent at 10x. And then you can do the 1x resequencing plus imputation of the whole progeny. And the third advantage is that I discussed with them and I said, okay, let's say we have a 1x resequenced accession and we find out that it's very interesting, we want to go up to 10x. Can you keep the plate and then retrieve the sample and send it for sequencing 10x, that particular sample? And they said yes. So this approach gives us the flexibility that we send the DNA once, they do 1x plus imputation, and if some of the accessions are very, very interesting, we go up to 10x with the same exact sample. That saves a lot of money and effort and everything. And then the third density is low density. We still have to decide how much low density is low density. From what I understood, 384 is not perfect. We have to develop a better panel, probably around 1000. So those would be the three ones. And based on this reasoning, my proposal is that the pools, the F2 population pools go to 10x. The whole Tradidom core collection goes to 10x resequencing. Before it was divided between 5x and 20x. My proposal is we do everything at 10x. I don't know what this HT donor is from SICK, but you asked for 20x. I propose we do 10x. And semi-ortho, 10x. The accessions, I think that's Serasiforme collection, right, Mathilde? The INRAE?
Yes, that's it. That's the end of the core collection for GWAS.
Okay, so that's also 10x. And so if you just want to to have haplotypes, the best thing to do is 10x. If you just want to do GWAS, 1x plus imputation could be also sufficient. That's your call. Then I don't know what this diverse material is, but before you asked for 5x, I moved it to 10x, and same for the N-Sazarian accessions is 10x. Then all the progenies from Huji, from what I understood, Danny wants to find breakpoints to finally map breakpoints. And for that, since 1x resequencing plus imputation gives you tens of thousands of SNPs all over the genome, I think that should be sufficient. What do you think, Danny? Is Danny around? No. Okay, I will ask him. Then we have some IELs here, intraspecific from SICK. I proposed, because there's dense genotyping, so this is 1x resequencing plus imputation is considered dense genotyping. So I propose we do the 1x, and the same for the MAGIC population. So that should give you, if I did correctly, the calculations, that should give you sub-centimorgan mapping of the breakpoints. We have to resequence the parentals first and then make a final decision. Maybe 1x will not be enough. Maybe you need a little bit more.
The MAGIC population parents have already been resequenced.
Okay, then you can have a look and you realize how much polymorphism.
Yeah, there is quite a lot of polymorphism because 4 parents are Pimpinellifolium and 4 are Ceraciforme.
Yeah.
So probably 1x is enough. But
yeah, I would start from there. You know what, Jaime? I would start with 1x plus imputation. Then you see if the data are satisfactory, you stop there for the moment. If they are not, you make use of this flexibility that they give us, and you go up to, let's say, 5x or 10x. What do you think?
Yeah.
Yes, I have talked with our colleagues, Santi, which is expertise in genotyping, and he told that this is enough. What you propose.
Yeah. Okay.
So for now, it is okay.
And as I said, go higher if there is the need.
Yeah, I think one additional question is, in the case of MAGIC or in the integration line population, the parentals are already sequenced basically to 10, 20-fold or more.
Sorry, there's somebody that is speaking in the microphone. Maybe Simo can mute him.
No, it's somebody from the same place. No, I was suggesting that in those cases that the parental lines have been sequenced to extensively, maybe because for the core collection, of course, is good to have them tenfold or more. But for the integration lines, it's okay, 1/4, because we think we can have the Pimpinellifolium sequence to high quality, but we still need to do, no, Simo, you suggested to do TO937 PacBio on top of what you already have. Is that correct? Simo?
Yes, it's correct. Yeah, we already did. It's the sequencing of the Pinelifolium T037.
Is it on this panel here?
Yeah, it's the parental line of the introgression and line population.
We did Oxford nanopore, but it's still missing some regions.
It's this one here.
No.
It's not here as a line, but it's the parental of this, the row 10. Specific IL specific. There's 54 lines are derived from the cross of the TO937 by Moneymaker.
Yeah.
Okay, so do you agree that we do this one by 1x plus imputation?
Yes.
Okay, good. Now for the parental.
But we have IC of this genome. I think we have 100x of Illumina that coming from HiC. If it is useful, we can send you the sequence.
The parental.
The parental, yes.
The parental. Okay, yeah, maybe you can. The parental will be fine. Yes. Yeah, I mean, if you have 100x, that's plenty.
Yes, but it's a contact map, so you can have when there is contact, the coverage is higher, as you know. But
but
sure. It covers very well the genome. There is no problem, and it works very well for the scaffolding.
Sequencing one more parental, 90 euros is not much. So whatever, it's not a problem. What I'm worried here is these guys here, that they have very high numbers, you know. And these ones, if you start doing at 10x or 5x, it's a lot of money. And as I said, you have the flexibility. You do them at 1x plus imputation. If you're not satisfied, you go higher. And here, Unina, I don't know what these are. I think this is Amalia. So I don't know what she's 6 genotypes for stress, but what is it? Is mapping or what?
Well, I think that the regional genotype that resulted resistant to its stress.
Yeah, no, but why does she need to sequence them? It's a mapping, or she wants to have the detailed sequence? What does she need it for?
I think that she wants to have details on the sequence.
And then, so this has to go to 10x.
Yes.
And this becomes 90. Okay. So that sounds.
Giovanni, I missed the beginning of the conversation because I was talking about person. I think for the pools, I will go to 20x.
Pools, you think
because 10x could be I mean, we lose resolution. I think we need 20x for the pools.
Okay. 20x. No, I understand if they are pools, it's not an individual accession, then you're right. Although you expect, you know, if a mutation is present in all the things, it should be the depth of coverage should be the same, right?
Yeah, but it is QTL, so probably will have frequency, different frequency, not all back and all one.
Okay. Let's do 20x.
Yeah. We sent Paola an update number, so we change a little bit the numbers, but it's not. Numbers are not exactly like this, but will be very similar.
Yeah. Okay, so, and then we have basically to work on the low density, because here, I mean, Andrea already told me he's not very happy with just having just 384. What about UPV, INRAE, etc.
For INRAE, also 384, I checked the parents with the list of SNPs that you sent. And actually, I have about only 30% which are polymorphic, so it's too low.
Yeah, I mean, that's a problem, right?
Yes, yeah. We have either to go through 1,000 if there are soil cap markers, or to have some specific ones.
Okay, so you know what we'll do. You know what we'll do. Is Giuseppe on the call?
Yes, yes, he is.
Giuseppe, can you activate your microphone?
Normally it's
Can you
activated.
hear me now?
Yeah, it's activated. Yeah.
Okay, good. So, Giuseppe, do you think we can take our 6 million SNPs and find those that have conserved regions left and right and that can be easily transformed into amplicon SNPs? Like you did for the 24 SNPs of the very low density, you remember?
Yes, we have to, yes, we can try.
We can try. So what we'll do, you know, we'll take the SNPs from the G2P soil collection. We'll select the ones that have conserved regions left and right and can be easily transformed into amplicon SNPs. And then we'll attach MAFs to them in our collection, so that minor allele frequencies, so that you can see, I mean, there are some that are very, very minor allele frequencies very low, so you want SNPs that have a good chance to be polymorphic. And then we'll come up with a list of those and send them to you. And ideally, they should be also located on genes, right? That's preferable. So after applying all these filters, we will send them to you so that you choose the ones that are more look better. What about that?
Yes, I think that's right. And maybe going, how many would you plan to do at that level? Still a new set of 380 or 1,000?
Okay, if you go back to my presentation, here it is. So the price for 384 SNPs is $5, including extraction, DNA extraction. Okay, for 1,000 SNPs, it's $9.50. So it does not.
How much? Sorry?
$9.50 per sample.
Okay.
So I would go somewhere in between 384 and 1,000. I would go for, maybe for the test run, I would do 1,000 and then take the ones that work really well, because, you know, sometimes you design them, but nevertheless, the primers don't work very well. So you have to aim to shoot a little bit higher to get, you lose some.
And also maybe to look at their distribution at the genetic level, not only at the physical level, because all the, for genetic mapping, all the centromere is not really interesting. We need very few SNPs in these regions.
So these 384 are equally distributed on heterochromatin and euchromatin?
I don't know. I didn't check.
I checked roughly. They are well distributed into chromosomes, so I suppose that they were also distributed along these chromosomes. But
Yes, but no, because they were derived from SOLCAP, and SOLCAP SNPs are usually more in genes, more frequent in genes, because they were derived from EST's sequencing. So maybe they are more numerous in the.
But we can have a look to that, so we can maybe if they are really evenly distributed, we can keep them.
They were not, but I don't think so.
Okay, so what we can do is, I mean, we'll, as I said, we will take SNPs that are in genes and that have conserved regions around them, and that's probably the best way will be the ones that are in introns and that have exons, just exons left and right. And that have a reasonable minor allele frequency so that they are informative.
Yes.
Then we send them to you guys and you tell us, okay, we would like this, this, and this. If we can reuse some of the ones in the 384 panel, that saves us money because each new SNP that we set up is $20. So let's say if we do a de novo panel of 1,000 SNPs, it's $20,000. If we do a de novo panel of just 200 SNPs and we add them to the 384 that we already have, that's much less. See what I mean?
Yeah.
Yes.
Okay. So we make the calculation together. We don't need to discuss this in detail now.
But Giovanni, for some activities, some people may need the data now or in a few months or in 1 year. So we need to also to introduce this piece of information, because if we need to do the 10 or 20 X sequencing all together or in separate times. I mean, how is going to be the process?
The timing, yeah, that's the timing. That's one important thing,
because it's important.
I see that the majority of INRAE and UNITUS were planned to be ready for June, July 21.
For resequencing, actually, for the low densities, it's a mistake. They will not be available before 22.
Okay. Okay. So
yeah, our plans are ready. I mean, we got the material already.
So maybe we can resend you this table or the ones that you corrected, but with the final good date.
With the final dates, yes.
Yes.
In case of PESIC and ENSA, there are some genotypes that we need as soon as possible, because we, depending of the genotype, we for example, for
the resequencing, right?
Yeah. So the pools, we need as soon as possible the pools, because depending on the results of the pools analysis, we'll have a different experiment in the year. So we have to choose. We need the results to decide the experiment for the year, and to introduce QTL, we need the resequencing of the ENSA parents to develop the markers.
Okay, I will send this table again.
Okay.
And you will tell me when do you want it. You're right, now.
We are close to Christmas now, so we can ask.
1 month ago.
We'll do our best to.
Okay.
But please, not everybody right now,
No.
because then it will be sort of difficult. Okay.
But is it better for you that you provide all the DNA at once, or for resequencing, or do you prefer to have it spread on time?
No, it's better. I mean, for 1X resequencing, the deal is that we send at least 96 samples every time and probably more. We'll try to send in shipments of 384 samples. Okay.
And for 10x, the same?
For 10x, maybe we can send less, but I wouldn't send 10 samples every time. I would try to group.
And do you have some specific conditions for DNA extraction, or?
Yes, Paula sent them already.
Okay.
It's either QIAGEN or LGC, this BDEX. These two protocols work really nicely. And if you don't want to extract DNA yourselves, there is a Dutch company that does automated extraction with LGC. It's relatively cheap. We can send you the address.
Okay, yes, please send it in case we are lazy.
And I think they ask €1 per sample, like that.
€1 per sample, so we send. We are not lazy, we send.
We can send. So.
It's more expensive.
It's more expensive for the post office.
But don't tell me that the labor costs in the Netherlands are falling that fast. This must not be labor cost. Must be a machine or a postdoc from another country.
All my DNA. Exactly.
I don't know what they do.
Any Dutch, yeah, there are Dutch here. But anyway, this is true.
Is Paula on the call? Paula remembers better.
Yeah, yes, I remember, but I remember also that we had a problem with the shipment of those samples. So the extraction was good, but when they ship the sample to us, they didn't do properly. And so we have to be careful about the shipping.
Yeah, I mean, we have to teach them how to seal the plates. I don't think it's a very complicated thing. They sealed the plates with the plastic film, and the film detached, and when the samples arrived, they were all mixed up. But if you use aluminum foil, then it.
Okay, is anything else, or we just wrap up and say goodbye? So, thank you all.
We have to go for the dinner. Yeah.
We have to go to dinner.
At 8.
Where will we meet?
Nearby. Yeah, please you may come.
So just hope that the next time we can see each other and just share the time, not just through the screen or the Canon. So it was very nice to see you all. And I think it has been a very constructive meeting. I think we have addressed every single issue. If not, we have at least the chance. So thanks a lot. So maybe Simo, you can take the last picture and then.
Okay, let's go. So maybe we can wait a little bit for people to switch on their camera.
Ahora.
Okay. A few seconds.
Okay.
Just 2 more seconds. Okay, so I will count until 3, and we will try to make a good smile at lunchtime for people that are in Europe. 1, 2, 3. And voila, first one and second one. Good.
Okay. Thank you.
Thank you. Thank you.
I'll see you soon. Thank you.
Bye.
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 Bye.